If you're studying for your AP Bio Semester 1 final and you're feeling anxious or nervous, that makes so much sense. AP Bio Semester 1 includes some tough topics, but don't worry. In this video, we're going to teach you everything that you need to know to crush that test. We're going to start with Unit 1, Biochemistry. We'll cover Unit 2, Cell Structure and Function. Unit 3, Cellular Energetics, including Cellular Respiration and Photosynthesis. Unit 4, Cell Communication, Feedback and the Cell Cycle. And Unit 5, Heredity. My name is glennwolkenfeld, also known as Mr. W, and I love teaching biology. To help you study, I put together a checklist. It includes everything that you need to get ready to study for this test. You can download it at apbiosuccess.com/checklist. AP Bio Unit 1 is the foundation for all of the great biology that you'll be learning this year. Unit 1 focuses on the chemistry and properties of water and hydrogen bonding, the elements of life, and the 4 biomolecule families that all living things are made of: carbohydrates, lipids, proteins, and the nucleic acids, DNA and RNA. To help you study for this Unit 1 test or the AP Bio exam, I've put together a checklist that you can download at apbiosuccess.com/checklist. Let's go. Water and hydrogen bonding. First thing to note is that water is a polar molecule. There's unequal electron sharing between oxygen and hydrogen. And so there's a partial negative region over here. There's partial positive regions over here. Note that hydrogen bonds are intermolecular bonds. They're between molecules, not within molecules like covalent bonds or ionic bonds or anything like that. So what's happening is that the oxygen is partially negative, the hydrogen is partially positive, and the hydrogen bond is a weak bond that forms between those two areas. Hydrogen bonds are much weaker, much weaker than covalent bonds, ionic bonds, any of those intramolecular bonds. Using the diagram below as an example, describe how hydrogen bonds can form between molecules besides water. The key idea is that hydrogen bonds are everywhere. They're not just within water. They're essential in biology in general. So in this example, there are 2 hydrogen bonds, and here's one oxygen to hydrogen over here. Here's another one between nitrogen and hydrogen. This is between the nitrogenous base adenine and thymine. And these are the hydrogen bonds that hold together DNA. Hydrogen bonds, again, are essential. They're key to the structure of DNA, RNA, proteins. You'll meet them again and again throughout your biology course, and they're important to know for the AP Bio exam as well. So now let's look at some of the consequences. So describe cohesion, adhesion, and surface tension, and explain how these key properties of water result from hydrogen bonding. Cohesion, that's hydrogen bonds between water molecules. So this is cohesion right here. And it's responsible for many of water's very peculiar properties. It's a very small molecule, but it has a very high heat of vaporization. It takes a lot of energy to get water to evaporate, a high specific heat. It can hold a lot of heat, and it has high surface tension, which we'll talk about below. Adhesion is water sticking to other stuff. So like, for example, here you see hydrogen bonds between water molecule and the cellulose walls that make up the conductive tubes of plants, which are also called xylem. A phenomenal thing is called transpiration, which is how water gets pulled up to the top of trees, and that's all based on water's ability to cohere to other water molecules and to adhere to the sides of the conductive tubes of plants as water evaporates from the top. And finally, surface tension. Here we see a paperclip that's resting on a net of water molecules, and of course that's ridiculously out of scale. You can see how that's keeping this paperclip from sinking down, and here it's the force exerted by water molecules on the surface of a body of water creates a kind of web or net upon the surface. In terms of hydrogen ions, hydroxide ions and pH, describe the difference between an acidic and a basic solution. The first thing to note is that acidic solutions are solutions that have more hydrogen ions, here's a hydrogen ion over here, than hydroxide ion. That's represented by OH over here. So if you dissolve this in water, you wind up having lots of hydrogen ions, and that pushes the pH down, all right? The pH is below 7. In bases, there are more hydroxide ions than hydrogen ions. So if you dissolve sodium hydroxide in water, you wind up with a lot more hydroxide ions, and that pushes the pH in this direction. pH is above 7. And one thing that I want to emphasize is that you don't really need to know about pH for the AP exam directly. There won't be a question about it, but it might be part of an FRQ or a multiple choice question. So it's an essential underlying concept. Elements of life, topic 1.2 of AP biology. So what do you need to know about the elements of life? Carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, sometimes referred to as CHNOPS. Carbon is the central element in all of the molecules that make up living things. Hydrogen, often used in energy exchange. We have molecules like NAD and NADH, which show up in cellular respiration. NAD plus is the low energy form, NADH is the high energy form. You see stuff like that over and over again. And also, hydrogen as an ion, as a hydrogen ion, a proton, is often pumped around to create energy gradients. It's very important in the synthesis of ATP. It's the basis of acidity and alkalinity, which we just talked about. But the main thing is to know about these atoms in context. Phosphorus is in phosphate groups, which is found in ATP. That's the kind of interconnected cross-topic knowledge that you want to have for success in AP Bio. Now on to topic 1.3, monomers and polymers and functional groups. So what are monomers? What are polymers? The basic idea is that 3 of the 4 groups of biomolecules, carbohydrates, proteins, nucleic acids, are built from smaller building blocks that are called monomers. So here's a glucose monomer over here. Living things build macromolecules, the big molecules, proteins, nucleic acids, polysaccharides, that have specific 3-dimensional shapes, and shape determines function, by combining these monomers into polymers. And a great analogy, if you hadn't already thought of it, is that the monomers are like Legos. You can combine them in any way to create other structures. And these big structures, the Millennium Falcon that you might have built as a kid or something like that, those are the polymers. And here's a note about structural formulas. If you see something like this, you might wonder, like, what's going on at these angles? Every unspecified angle vertex has a carbon atom. And that's why this is a C6H12O6, because there's carbons here, 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 here. Carbon is so central that it's just understood. How do you put monomers together to make polymers? It's a process that's called dehydration synthesis. Everything in biology is run by enzymes, as we'll see in Unit 3. And what enzymes do is they pull a hydroxyl group over here off one monomer, and it pulls a hydrogen off the other one. So here's the hydroxyl, here's the hydrogen. That water is pulled out, right? Because this is H2O, the water gets removed, and that creates a bond that's right over here. So here we see that between 2 amino acids, and same thing. So 2 monomers becoming a larger molecule. Dehydration, synthesis, easy to remember. Synthesis is how you build things, and dehydration, when you're dehydrated, you lack water. So dehydration synthesis is pulling out a water. Now, what about hydrolysis? Hydrolysis is the opposite of dehydration synthesis. In biology, the suffix "lyse" is very important, and it involves breaking. So what happens in hydrolysis is that an enzyme, which isn't shown, inserts a water molecule in between the 2 monomers making up the polymer. And what that does is it breaks them apart. Here we have lactose, which is a disaccharide. I'll review that in a minute. It's a sugar that's made of 2 simple sugars that are bonded together. You add a water molecule and you get galactose and glucose. What do you need to know about functional groups? In one sense, not a lot. They're not going to directly appear on the AP Bio exam. But in another sense, they're very important in terms of you decoding what's happening with the molecules in biology, which means the molecules that might appear on the AP Bio exam or in your course. So let's go through a couple. Phosphate groups, number 1 over here, they're key for energy exchange. ATP, adenosine phosphate. Phosphates are also found in DNA, and they're what energize DNA monomers as they're put together. This methyl group over here at number 2, it's used to silence DNA. It makes molecules non-polar or hydrophobic. There are a couple of polar functional groups to know about. There's the hydroxyl group at 5, the carbonyl group at 7, and they make molecules hydrophilic or water soluble. The carboxyl group, that's at number 3, and the amino group, that's at number 4, those are essential in amino acids, which obviously have an amino group over here, have a carboxyl group. The sulfhydryl group over here at number 6 is very important in terms of protein structure. It creates a stabilizing bond that holds proteins in a specific three-dimensional shape. And the acetyl group at 8 is used to activate DNA through a process called acetylation. So it's kind of the opposite in terms of function from the methyl group that we talked about. Carbohydrates and lipids. So what are the 4 types of macromolecules that make up living organisms? What can you identify from this diagram? So what you have here are carbohydrates, represented by this disaccharide over here. We have a lipid, represented by a phospholipid, a key molecule in membranes. We have a protein represented by hemoglobin, and a nucleic acid represented by DNA. So those are the big 4. What do you need to know about carbohydrates? Carbohydrates, the monomer, monosaccharides, those are simple sugars, and some of those are all important in biology, like for example, glucose, which is essentially the fuel of life. Disaccharides are going to show up less in the course, but you might have a question that's about lactose and lactose intolerance. Well, it really doesn't make sense unless you know that lactose is a disaccharide composed of 2 linked monosaccharides. And then you have polysaccharides, molecules that are used for energy storage, like starch, which is in plants, glycogen, which is in you and other animals, and then polysaccharides that play important structural roles, like cellulose, which makes up the cell walls of plants. While humans and other animals can't digest cellulose for food energy, a few animals such as ruminants and termites can. Explain. Most animals can't hydrolyze the bonds that connect glucose monomers in cellulose. So here's cellulose, it's a polysaccharide, it's a bunch of linked glucoses, but it's linked in a way so that you don't have the enzymes that can break this bond, freeing up the glucose monomers. So essentially, you could eat lettuce, celery, these are high cellulose foods all day. You'd never get enough calories to really power your life processes. You do have the enzymes that can break this bond in starch. So over here, and over here, and over here, and that enables you to convert starch into glucose, and that enables you to use that glucose to power cellular respiration. There are a couple of animals, such as termites and ruminants. Ruminants include cows, sheep, goats, deer, other animals, other mammals. And what they did is they involved symbiotic relationships with microorganisms that can hydrolyze this bond and thereby break this bond over here, break this bond over here, break this bond over here. And that releases these glucose monomers, making food energy available. Let's end our review of carbohydrates by looking at an issue in relatively recent human evolution related to carbohydrates. And that's about the biology of lactose tolerance and intolerance. So here's what you need to know. Lactose is the sugar in milk. Here it is, it's a disaccharide. Lactase is the enzyme that hydrolyzes lactose into monosaccharides. And here you see that reaction happening. Most mammals only produce lactase during infancy while they're suckling, because that's the only time that most mammals ever drink milk. So that makes sense from an evolutionary point of view, because when you're an adult, you don't produce lactase. Why should you produce an enzyme for something that you don't eat? But what happened in human evolution is that some human groups that were pastoralists, herders, these are people who were associated with cows and sheep and goats and so on and so forth. They had access to all these milk products from the cows and the sheep and the goats, and some of them developed a mutation that enabled them to continue to produce the lactase enzyme into adulthood. And that opened up a whole niche of food exploitation that wasn't otherwise available. Now, this didn't happen all over the world. This happened in a couple of hotspots. Here's one in Africa, here's one in Europe, and here's one in Saudi Arabia, current Saudi Arabia, and here's one that is in the Indian subcontinent. And in those areas, lactase persistence, production of lactase into adulthood, became widespread. But in large areas of the world, that never happened. And there are many humans, the majority of humans, who are lactose intolerant as adults. But if that's true of you, as it is of me, then we have products like Lactaid. Lactaid is essentially lactase, the enzyme that you can use as an additive to food, and it'll break lactose down into galactose and glucose, and you can buy Lactaid milk and so on and so forth, and that's how that problem, if it's even a problem, is solved. Now let's move on to lipids. Let's talk about lipids and what are their functions. So here we have 4 different lipids, and what makes a lipid a lipid? First of all, lipids are molecules that are wholly or partly non-polar. So for example, like you see all these hydrocarbons over here, those are all completely non-polar. They don't dissolve in water, they're hydrophobic. They also are characterized by the fact that unlike the carbohydrates that we've met and the proteins and the nucleic acids that we will meet, they're not composed of repeating monomers. They might have subunits, but they don't have those subunits repeated hundreds or dozens of times. So what are their functions? Well, over here we have a fat, a triglyceride, and that's used for energy storage. That's true in both animals and plants. In animals, those fats are usually solid. In plants, those fats are usually liquid. We call them oils. Here's a wax. Waxes are used for waterproofing. This molecule is a phospholipid. It makes up cell membranes. We'll talk about that later. And then we have a steroid hormone like estrogen or testosterone that's used for signaling. So what's the relationship between phospholipid structure and membrane structure? Well, here's what you need to know. Phospholipids have a hydrophobic non-polar tail. That's this over here. And they have a hydrophilic or polar head, and those two parts are connected by a molecule of glycerol. And the key thing is that when you mix this kind of molecule in water, they spontaneously form an orientation where the heads will interact with water and the tails will avoid water. Hydrophilic over here, hydrophobic over here. And so if you can think about that arranged in a kind of spherical way, you wind up having a structure that is a bilayer, 2 layers of phospholipids. And that's the structural framework of cell membranes. We'll talk much more about that when we do Unit 2. At learn-biology.com, we understand why students struggle with AP Bio. It's a hard course. The material is complex, the amount of vocabulary is ridiculous, and the pace is withering. It's natural to feel overwhelmed and inadequate. To get an A or a 4 or 5, you need an easier way to study, and that's why we created learn-biology.com. It's got quizzes, it's got flashcards, it's got interactive tutorials about every topic in the AP Bio curriculum. It's got a comprehensive AP Bio exam review system. Use learn-biology.com and you'll gain the skills and confidence to ace your course and to crush it on the AP Bio exam. Here's your plan. Go to learn-biology.com. We've got free trials from June through March for both teachers and students. Sign up. You won't believe how much you'll learn. Let's review proteins. So the monomer is an amino acid, and it has a central carbon over here, and connected to that carbon is an amine group over here that makes this basic in its structure, but over here on the other side it's got a carboxyl group and that makes it acidic, so therefore it's an amino acid. There's a hydrogen atom attached to the central carbon, and then there's a variable group or an R group, and there are 20 variations, and that's true of all life. All life is built of the same 20 amino acids. That R group is also called a side chain, and it can be polar, nonpolar, acidic, or basic. There are 4 levels of protein structure. This is a super important topic. We're going to do an overview and then we're going to walk through all 4 of these levels. Primary structure is what's shown over here in A. It's a linear sequence of amino acids. It's genetically determined. The secondary structure, which is shown here and here, those are interactions that involve what's called the polypeptide backbone. When you do a dehydration synthesis and connect one amino acid to the next, to the next, to the next, that chain of carbon-carbon-nitrogen, carbon-carbon-nitrogen, that's the polypeptide backbone. The next level is called tertiary structure, and those are interactions between those R groups. And then finally, there's quaternary, fourth level structure, and that involves interactions between multiple folded tertiary peptides. Okay, let's talk about primary structure. In this diagram, A1, A2, A3, those all represent different amino acids. So the sequence of amino acids that make up a polypeptide, that's what you call multiple amino acids linked together, that's the primary structure. Proteins aren't really built by enzymes in the way that everything else is. They're built by ribosomes. The amino acid, here's one, here's another one, they're connected to one another by peptide bonds. So I was saying before, nitrogen, carbon, carbon, nitrogen, carbon, carbon. That's the polypeptide backbone. All of those amino acids linked together. That's primary structure. What's secondary structure? Well, here you have a nice diagram that shows you what that polypeptide backbone is. And the secondary structure emerges as interactions between the carbonyl groups over here and the amino or amine groups over here within the polypeptide backbone. Now, what happens is that interactions between these amine groups and these carbonyl groups, they form hydrogen bonds and they stabilize certain shapes. One of the shapes to know about is called an alpha helix, and that's kind of a corkscrew over here. So you can see that there's a hydrogen bond that's stabilizing this, hydrogen bond, hydrogen bond. So that forms that shape. Now the other thing that happens is if the parts of the polypeptide chain are either parallel to one another, like this, or anti-parallel to one another, like this, then carbonyl and amino groups can again interact and form hydrogen bonds, and that can lead to a form called a pleated sheet. And that's what we see over here. Tertiary protein involves interactions between the side chains or the R groups. And there are a couple to know about. First of all, there are hydrogen bonds, shown at number 2. There are ionic bonds that are shown at number 5 over here. There are covalent bonds, which are shown at number 3. That was between two sulfhydryl groups, another one of the functional groups. And this is a covalent bond that's very important in really tightly holding that protein into a specific shape. And then finally, you have what's called hydrophobic clustering, where non-polar side chains will cluster together, avoiding water. Down here, you see myoglobin, which is an oxygen-storing protein that's found within muscle tissue. That's a tertiary protein folded into a specific shape. And you can see, like over here, there's a whole bunch of alpha helices that are in that structure. Quaternary structure involves multiple polypeptides that interact with one another to create the final form of the protein. So those interactions might be hydrogen bonds, they might be ionic bonds, they might be hydrophobic interaction. In this diagram, actually, you can see all 4 of the levels. So here's the primary structure. Here's an alpha helix secondary structure. This hairpin turn is actually part of the tertiary structure over here. And then you have multiple polypeptides interacting. This molecule looks a lot like hemoglobin. This is kind of cool in light of our recent history. The spike protein that's on the outside of SARS-CoV-2, and in fact all the SARS viruses, is also a quaternary protein that's made of multiple folded polypeptide chains. An application of what we learned about proteins is this question. Describe the structure and function of hemoglobin and explain the molecular cause of sickle cell disease. So sickle cell disease is an inherited blood disorder. It was one of the first molecular genetic diseases that was really well understood. The molecule that is in question here is hemoglobin. And hemoglobin, its function is to transport oxygen in our red blood cells. The structure, it's a quaternary protein. It's made of 4 polypeptide chains. Sickle cell disease is caused by a recessive mutation. We'll cover that in Unit 5. But The key idea is that there's a mutation that causes the amino acid valine over here to substitute for glutamic acid. And that's an important mutation because glutamic acid is acidic, whereas valine is non-polar. The result of that is that when blood becomes deoxygenated, those mutated hemoglobin molecules, they form hydrophobic bonds with one another. Well, why? Because they have a hydrophobic amino acid sticking on the outside, and that causes them to do this. They cause fibers to develop within the cells, and that causes the cells to become spiked like this. They're mutant cells, and those cells will then clump up within smaller arteries, and that causes these pain crises, it causes tissue damage. It's a debilitating disease. There are a few other things to know about sickle cell anemia. The first is that during the 30 years that I've been a biology educator, sickle cell disease has been transformed from a childhood disease with certain early mortality to a disease that can be managed with good medical care so that people can live into their 50s, 60s, and even beyond. Sickle cell disease is also a target for gene therapy, something that we'll talk about in Unit 6. And finally, there's a really interesting evolutionary thing where sickle cell disease evolved because having one copy of the gene is actually a benefit that gives you resistance to the tropical mosquito-borne disease, malaria, something that we'll talk about in Unit 7. And now for our last topic in this Unit 1 review, nucleic acids, DNA, and RNA. Note that we'll give an overview now, and we come back to nucleic acids when we cover gene expression in Unit 6. So we'll start by describing the biological importance of the nucleic acids. These are the molecules of genetic information. DNA is the molecule of heredity. It's what passes from generation to generation. It's the molecule that cells pass on as they divide and replicate within a multicellular organism. RNA has other functions. RNA is the hereditary molecule in some viruses, never in cells. And RNA's key role is information transfer, as in messenger RNA. So within a cell, here is DNA, the repository of genetic information. It has its information transcribed into RNA, and then that RNA goes into the cytoplasm where a ribosome will translate that RNA message into protein. RNA is a very versatile molecule. It's not in the form of a double helix the way DNA is. It can take many, many forms, and it can act as an enzyme catalyzing reactions. Ribosomes are essentially catalytic RNA. There are also other molecules called spliceosomes, microRNAs, that play a whole variety of functions. And I finally want to say that this typically isn't put into this unit, but ATP is a nucleotide monomer. It's one of the monomers of RNA, and it's the energy molecule of life. It's how cells get work done. What is the monomer of nucleotides? What's its structure? How are these monomers different in DNA and RNA? The monomers are called nucleotides. We just looked at ATP. All of these molecules have a 5-carbon sugar that's shown at number 2. They have a phosphate group that's shown over here at number 1. And then there's one of 4 nitrogenous bases. So the nitrogenous base doesn't have to have this structure. Note that the phosphate group is connected to a number 5 carbon, whereas the nitrogenous base is connected to the number 1 carbon. When you learn about DNA replication, you'll talk about things like DNA is replicated in a 5-to-3 orientation. And now you know that's about the 5 carbon over here and the 3 carbon over here. In DNA, the sugar is deoxyribose, and there are 4 bases: adenine, thymine, cytosine, and guanine. In RNA, the sugar is ribose, and the bases are adenine, uracil, cytosine, and guanine. So now let's talk about the structure of DNA. DNA consists of 2 nucleotide strands, so easier to see in this flattened out version. Here's one, here's the other. Within each strand, the nucleotides are connected by sugar-phosphate bonds. So here's a sugar, here's a phosphate, here's a sugar, here's a phosphate. But the strands connect to one another by hydrogen bonds. So here you see G connecting with C, A connecting with T, and those are rules to memorize. Adenine, A always bonds with T, C bonds with G. They have complementary shape. They're molecular dimensions are such so that they fit nicely within the helix. That's more of a story for Unit 6. And note that these two strands fit together in an anti-parallel orientation. In order for the nucleotides to form hydrogen bonds with one another, they each need to be upside down relative to the other. And that's what anti-parallel is all about. We'll talk about this again more in Unit 6, but just to lay this down for right now, DNA is directional. Notice that in a chain of nucleotides, these are RNA nucleotides, you can tell because there's uracil. The nucleotide sugar binds with a phosphate, and then there's a sugar, there's a phosphate, there's a sugar, there's a phosphate. Well, the enzymes that build DNA, it's called DNA polymerase, and these enzymes, like all enzymes, have an active site. They only can work in certain orientations, and they work completely by feel. And they can only add new nucleotides at the 3' end of a growing nucleotide strand. So that's what directionality is all about. All of these nucleic acids are built in the 5' to 3' direction. If you find yourself looking at a diagram like this one of an animal cell and thinking to yourself, how am I going to master all of this material? If you're feeling overwhelmed and nervous, then this video is for you. Here's what we're going to cover. We're going to start with an introduction to cells, then we'll look at why cells are small. We'll follow that by looking at how cells are internally organized. That's about cell compartmentalization and a system of organelles that are organized into an endomembrane system. After we do that, we'll be able to walk through cell parts and functions. Then we'll look at membranes, how membranes are made up, how membranes transport things from one side to the other, and finally, we'll look at osmosis. My name is glennwolkenfeld, also known as Mr. W. I'm a retired AP biology teacher. I love biology. To help you study, I've put together a checklist that you can download at apbiosuccess.com/checklist. Cells and introduction. Describe the basic structure and fundamental parts of cells. Cells are the basic unit of life. They're the basic unit of structure and function in every organism. In terms of their own structure, they have a membrane which separates the cytoplasm, all the living material inside the cell, from the cell exterior. They always have genetic information in the form of DNA, the double helix, and there are systems that maintain and replicate the cell as a whole and the cell's genetic information, its DNA. One of the most important systems is a system by which the genes in DNA get transcribed into messenger RNA, another nucleic acid, and that messenger RNA is read by a particle called a ribosome and translated into proteins. Proteins are all important. They're the workhorse of the cell, and one of the most important proteins are enzymes. Enzymes control the cell's metabolism. Other proteins might be embedded in the cell's membrane, and others might even be exported from the cell. Compare and contrast prokaryotic and eukaryotic cells. Among living organisms, there's a great divide between the prokaryotes and the eukaryotes. Here's what it's all about. Prokaryotic cells are small and relatively simple. They have no nucleus. Their chromosome is circularized, and what that means is that there's a loop. The beginning and the end meet up. They contain extra chromosomal pieces of DNA that are called plasmids, and they're found in 2 of life's 3 domains, the archaea and the bacteria. Eukaryotic cells are larger and more complex. They're found in the domain that has their name, the eukarya. They have a nucleus. They have multiple linear chromosomes. So here they're shown over here, and you can see the chromosomes here. They have a beginning and an end. And those chromosomes have DNA that's associated with proteins, which is different from the simpler DNA packaging that is found in the prokaryotes. Eukaryotes have mitochondria, and the presence of that organelle is the defining feature of the eukaryotes. And they have many membrane-bound organelles. Membrane-bound means surrounded by a membrane. AP Bio topic 2.3, cell size, an essential topic. Use the relationship between surface area and volume to explain why cells are small. Cells need to have enough membrane surface area to allow for diffusion of substances and molecules in and out. Everything that gets into a cell gets there through the membrane. And it might be counterintuitive, but a small cell has much more surface area relative to its volume than a large cell does. Let's look at the math. If you have a cell that's 1 micrometer, 1 micron on the side, that's its length. Then its surface area, if it's cubic, would be 1 times 1 times 6. Think of the formula for surface area for a cube. Its volume is 1 times 1 times 1, and therefore its surface area to volume ratio is 6 to 1. 6 units of surface area to every 1 unit of volume. Now, if you have a larger cube, one that's 10 micrometers, then its surface area is 600. Wow, that seems like a lot. But its volume is 1,000 because volume's a cubic function and it's going up much faster. So if you look at the surface area to volume ratio, it's 0.6 to 1. As an object gets larger, its amount of surface area relative to its volume decreases. And in this example, the larger cell's surface area to volume ratio is 1/10 that of the smaller cell. And a large cell can't efficiently use diffusion to get the nutrients it needs in and to get waste, carbon dioxide and metabolic wastes out. Why do organisms need to increase surface area in certain tissues? How do they do that? The why is in order to increase the surface for diffusion of molecules or heat. How? You can have thin sheets of tissue, like these thin sheets that make up the gills of a fish that have been dissected out. That's for increasing the intake of oxygen and the diffusion out of carbon dioxide. Or if you think about the ears of elephants, those are big, flat sheets of tissue that enable the elephant, which has very little surface area relative to its volume and lives in a hot place, to get heat out of its body. You can also have highly folded surfaces, like the internal membrane of the mitochondria or the structures that are called villi that make up your intestine. And there's actually folds within folds. Explain in terms of surface area why there are no small mouse-sized marine mammals, and why some marine mammals, like the whales, the largest organisms to have ever evolved, are really huge. Mammals are warm-blooded. Ocean water is cold. Increased size decreases the surface area to volume ratio, so there's less heat lost to the environment. Think about a mouse. It's small. It has a lot of surface area relative to its volume. It would be over here on the graph. It couldn't survive because it would experience so much heat stress in the water. The smallest marine mammals are the otters, and they have a thick coat of fur that helps them maintain their body temperature. Whales have evolved to be larger and larger for a variety of reasons, but one of which is that by being big, they lose relatively less heat to the environment than a smaller animal would. It's all about the surface area to volume ratio. Cellular compartmentalization and the endomembrane system, how eukaryotic cells are organized. What is cell compartmentalization? What are its advantages? Compartmentalization means internal division of a space into sections. Think about the overhead luggage compartment in an airplane. What are the advantages? The first is that it allows a cell to have regions with an internal chemistry that are distinct from the cytoplasm. A great example of that is the lysosome, shown at G over here, and number 7 over here. Lysosomes contain hydrolytic enzymes. Those are enzymes that break things down. Those can't be released into the cytoplasm, or they would break down the cytoplasm, so they are compartmentalized. They're kept in a membrane-bound space. In addition, compartmentalization provides a lot of internal surface area, and that's important for membrane-bound enzymes that are found in the smoothie R or the Golgi that can only do their job when they're embedded in a membrane. Compartmentalization makes that possible. And that's also true of the ribosomes that are found in the rough ER. Compare compartmentalization in prokaryotic and eukaryotic cells. Prokaryotic cells have few compartments, though there are internal regions with specialized structures and functions. Here's a prokaryotic cell. You can see that its internal structure is much simpler than that of a eukaryotic cell, but one notable compartment is called a thylakoid, and it's this over here in a cyanobacterium. Cyanobacterium evolved into chloroplasts. We'll talk about that in a moment, and they have these internal compartments called thylakoids that are essential for the function of photosynthesis. Eukaryotic cells are highly compartmentalized. There are many internal membranes that divide the cell into regions with distinct structures, distinct chemistry, and functions. And examples include lysosomes, the endoplasmic reticulum, the Golgi complex, and vacuoles like this large vacuole in a plant cell. What is the endomembrane system? It's a dynamic connected system of internal membranes and compartments. It includes the nuclear membrane, the rough ER, the smooth ER, the Golgi, lysosomes, and all the little vesicles that interconnect them. Membrane and material flow from one compartment to the next. So the membrane that's part of the rough ER might become the same membrane, the same phospholipids that are part of the smooth ER, and those might become parts of vesicles which flow into the Golgi. So those phospholipid molecules are the same. It's dynamic, there's constant exchange. Explain the origin of mitochondria, cellular compartmentalization, and chloroplasts. What we're explaining is the origin of the eukaryotes. When did this happen? About 1.8 billion years ago. Before that, there was nothing on Earth but prokaryotes. How did this happen? It arose as a mutualistic endosymbiosis. Symbiosis means living together, two species living closely associated with one another. Endosymbiosis is when one of those species lives inside the other. We normally think of that as parasitism. That's a negative kind of endosymbiosis, but there's also positive endosymbiosis. That's a mutualistic relationship, and that is what gave rise to the eukaryotes. So what happened is that here we have representatives of the two prokaryotic domains, and an archaeal cell took up a bacterial cell. Now I want to emphasize that there are many scenarios and many diagrams that represent how this happens, and this one is the one that makes most sense to me. So the bacterial cell entered inside the archaeal cell and it evolved into a mitochondrion. That mitochondrion, still kind of the bacteria, secreted vesicles, and those vesicles became the nuclear membrane, shown over here, and it started to develop structures that are part of the endomembrane system, like the endoplasmic reticulum, which is shown over there. So that's how we have eukaryotes. Then later, a second endosymbiotic event led to chloroplasts, which were free-living cyanobacteria, entering into a eukaryotic cell, and that set the stage for algae and plants. What's the evidence that chloroplasts and mitochondria were once free-living bacterial cells that arose through endosymbiosis? What's the evidence for this idea? Like bacteria, both of these organelles, mitochondria and chloroplasts, have their own circular DNA. They replicate themselves through binary fission. That's how prokaryotes replicate themselves. They use their own ribosomes to produce some of their own proteins. These ribosomes resemble bacterial ribosomes in terms of their rRNA sequence and structure, and they have two membranes. The outer one, so the outer bacterial membrane, let's consider it over here, is a vestige of an endocytotic vesicle. At learn-biology.com, we understand why students struggle with AP Bio. It's a hard course. The material is complex, the vocabulary is ridiculous, and the pace is withering. It's natural to feel overwhelmed and inadequate. To get an A or a 4 or a 5, you need an easier way to study, and that's why we created learn-biology.com. It has quizzes, it has flashcards, it has interactive tutorials about every topic in the AP Bio curriculum. It has a comprehensive AP Bio exam review system. Use learn-biology.com and you'll gain the skills and confidence that you'll need to ace your biology course and to crush it on the AP Bio exam. So here's your plan. Go to learn-biology.com. We've got free trials from June through March for both teachers and students. You won't believe how much you'll learn. Eukaryotic cell parts and functions. Describe the structure and function of the nucleus. The nucleus's function is to store and protect genetic information in the form of DNA. That DNA is wrapped around proteins to form chromosomes. This representation here is only present during mitosis or meiosis when cells are dividing. Otherwise, the DNA is more diffuse and spread out. It's still in chromosomes, but we call the form chromatin. The nucleolus is this dark part in the center of the nucleus, and it assembles ribosomes. The entire nucleus is surrounded by a membrane that separates the chromosomes from the cytoplasm, and the nuclear membrane has pores that allow molecules to enter and leave the nucleus. Important among those are messenger RNA, which leaves the nucleus, goes into the cytoplasm, where it's transcribed into proteins, and various transcription factors that come into the nucleus to turn genes on or to turn them off. Describe the structure and function of ribosomes. Ribosomes are particles composed of ribosomal RNA and protein. They consist of a large subunit and a small subunit. Their function is to read a genetic message encoded in a sequence of messenger RNA and to translate that message into a sequence of amino acids that make up the primary structure of a protein. The details of translation, protein synthesis will occur in Unit 6. What are two locations within a eukaryotic cell where ribosomes can be found? Ribosomes can be free or bound. Free ribosomes, shown at 3, float freely within the cytoplasm, which is number 5. Bound ribosomes, shown at 4, are connected to the membrane of the rough ER. All ribosomes start out as free, though through a process called protein targeting, they migrate to the ER to become bound. They do that if the proteins that the ribosomes are creating are destined to be put inside a vesicle so they can go to the Golgi or the membrane or a lysosome. Describe the structure and function of mitochondria. Mitochondria are shown here in the context of an animal cell. Here's a larger image. The function is converting food energy into ATP. That's the molecule that cells use to get work done. The key structures, there's a chromosome that consists of DNA. There are also ribosomes, which aren't shown in this diagram. Evidence of endosymbiosis. There's an inner membrane. It's highly folded. That increases surface area. And that's because there are many membrane-embedded enzymes and proteins, one of which is shown at 5, that are involved in the process of ATP synthesis. The mitochondria, as a former independent cell, has its own cytoplasm. It's called the matrix. It has enzymes for the Krebs cycle and enzymes that perform other functions. There's an intermembrane space, which is an important cellular compartment that enables the mitochondrion to produce ATP. We'll see how that works in Unit 3. And there's an outer membrane that is a vestige of endosymbiosis. Describe the endoplasmic reticulum, the ER, and list its two forms. The ER is an interconnected series of channels found between the nuclear membrane over here and the Golgi body in eukaryotic cells. So here's the ER, and it consists of two forms. There's the rough ER and the smooth ER. Describe the structure and function of the rough and smooth ER. Well, the rough ER is studded with ribosomes. That's what all these little dots are over here. And using those ribosomes, the rough ER synthesizes proteins for inclusion in lysosomes, in other organelles, in the membrane, or for export from the cell. The smooth ER is on the outer side of the ER network, always towards the Golgi, and it lacks ribosomes, but that doesn't mean that there's nothing in that membrane. It has many membrane-embedded enzymes, and the functions of the smooth ER vary by tissue. So it can include synthesis of lipids, converting toxins into soluble forms that can be excreted from the body, carbohydrate breakdown, and synthesis. Describe the structure and function of the Golgi complex. The Golgi is a series of membrane-bound flattened sacs, and you'll remember that flattening increases surface area. The Golgi receives vesicles from the rough and smooth ER and chemically modifies their contents. So here's the rough ER, here's the smooth ER, here's the Golgi, and here's a vesicle bringing something from the ER system to the Golgi. The Golgi packages those modified proteins into vesicles, shown at 6, that are sent to organelles like lysosomes over here at 7, to the cell membrane, or exported from the cell. Describe the structure and function of lysosomes. Here's a lysosome over here, here's a lysosome over here, and one thing that I rarely say, but I'll let you know that you will almost certainly never have to identify a lysosome on a diagram on the AP Bio exam. It's just a big vesicle. Lysosomes are membrane-bound organelles that contain hydrolytic enzymes. They're only found in animal cells. They carry out intracellular digestion. So like, for example, here you see a particle. It actually looks like a cell that's being engulfed by the membrane. It'll be packaged into a vesicle, and that vesicle will be sent to a lysosome where hydrolytic enzymes will break this down. The lysosome also recycles worn out, damaged, or excess organelles and molecules, and it plays a key role in a process called apoptosis. The P is silent. That's programmed cell death, and we'll talk about that later in the course. The cytoskeleton is a dynamic network of protein fibers, and those fibers make up the bones, the skeleton of the cell. In the same way as our skeleton, our bones, working with muscles enables us to move, the cytoskeleton is a dynamic network that enables the cell to move both internally, so it enables cells to move materials and organelles within themselves, and it also enables the cell to move their membrane so that they can do things like endocytosis, where cells can engulf things by moving their membrane, or also moving their entire bodies. This is an organism called an amoeba, and it moves by extending parts of the membrane over the surface, enabling it to crawl. How does it do that? Through the cytoskeleton. What are centrosomes and centrioles? Centrosomes are the organelle. They contain 2 centrioles. And the function is creating spindle fibers, that's what these are over here, that separate chromosomes during mitosis and meiosis. The parts that we've talked about so far have all been represented within animal cells, but most of those parts are also found in plant cells. The one exception is the lysosome, unique to animal cells. Let's describe the function of the central vacuole, this large structure right here. It's only in plant cells. Its functions include water storage, storing and releasing macromolecules, sequestering waste products, and maintaining turgor pressure. We'll talk about this in the context of osmosis, the movement of water, but as water flows into a cell, it creates outward pressure that keeps plant cells full, firm, upright. They're not wilting. Describe the evolutionary origin and function of chloroplasts. Here are chloroplasts over here. Here's a single chloroplast. Chloroplasts are the endosymbiotic descendants of free-living photosynthetic bacteria, and that's why they have their own DNA, their own ribosome. That's why they have 2 membranes. Their function is creating carbohydrates through photosynthesis. We're going to leave their detailed structure and how they work for Unit 3 when we discuss photosynthesis. Describe the chemical composition and the function of the plant cell wall. The cell wall is composed primarily of cellulose, which is a polysaccharide. It's that polysaccharide that we can't digest, but which ruminants, cows, goats, can digest because they have these symbiotic relationships that enable them to break that down and release food energy. The major function of the cell wall is it acts as a pressure vessel that prevents overexpansion in response to inward flow of water, osmotic pressure. So here we see as water flows into a plant cell, the central vacuole will expand and it'll look like this. The cell will be full of water, and that water will push against the cell wall, keeping the plant cell full and firm, avoiding wilting. So that's a good thing for the plant. And the cell wall is the primary component of wood and the water conducting tubes in plant stems. Membrane structure and function. What is the function of the cell membrane? The cell membrane separates the highly organized contents of the cell from the cell's environment. Cells are open systems. They need to let things like food in. They need to let things like wastes out. The membrane is how that's controlled. That's because the membrane is a selectively permeable boundary. It allows the passage of some substances and particles through, but not others. That selectivity is really the key to life. Describe the structure of phospholipids and their role in cell membranes. Phospholipid structure, there's a hydrophobic nonpolar tail region. Here's the structural formula. Here's a representation. There's a hydrophilic polar head. Notice that the head has this phosphate group over here with its negative charge. The whole thing is held together by this 3-carbon molecule called glycerol. In solution, when phospholipids are mixed with water, the heads of the phospholipids bond with water molecules while the tails form a water-free zone. So interacting with water, water-free zone over here. That creates a bilayer. That's the basic framework of the membrane to which proteins and other molecules are added. We'll see that further on. And the phospholipid bilayer is stabilized by weak bonds, van der Waals bonds, that occur between the tails. Describe the fluid mosaic model of cell membranes. This model posits that the membrane is composed of phospholipids plus proteins plus cholesterol in motion. So why is it called fluid mosaic? Well, it's fluid because the components are moving around laterally within the plane of the phospholipid bilayer, which you can see here. It's a mosaic because it's composed of a variety of pieces in the same way as a mosaic tile is composed of different tiles with different colors that get put together to form the art. Within the phospholipid bilayer, you have phospholipids that are at 4 and 9. You have proteins that are at 1, 3, 5, and 6, and you have molecules like cholesterol, which are shown at 7. And then inside and outside the phospholipid bilayer, you might have additional molecules attached to proteins or phospholipids. And those might be glycoproteins, which are not shown, but at 8 you have a glycolipid. Glyco means it's a carbohydrate and it's attached to these fatty acids right over here. Describe how proteins fit into the cell membrane. There are 3 ways that proteins can fit in, and if you think about hydrophobic, hydrophilic, what binds with what, it makes a lot of sense. So there are transmembrane proteins. That's shown over here and over here. Here, notice that beautiful alpha helix that you should remember from our Unit 1 review. There's a hydrophobic core that fits into the nonpolar inner portion of the membrane. So these would be hydrophobic amino acids over here and over here. And there'd be hydrophilic regions that extend into the cytoplasm and the membrane exterior. There are integral proteins, such as the one at 5, and these proteins would have a nonpolar region that extends into the hydrophobic membrane middle, and a single hydrophilic region that juts into the cytoplasm, as this one does, or into the cell exterior. And then finally, you have peripheral proteins, like this one that's shown at 3, and they attach to phospholipid heads that are either on the cytoplasmic side of the membrane or in the cell exterior. These terms, transmembrane, integral, peripheral, they're not important in and of themselves, but you should be able to understand the concept of how things fit into the membrane. Membrane transport, how things get across the cell membrane. Diffusion is the tendency of molecules to spread out from where they're more concentrated to where they're less concentrated. So here you have some substance more concentrated over here, less concentrated over here. Here's ink going into a beaker, more concentrated, and then less concentrated. This happens spontaneously. It's based on the kinetic energy of the molecules. And a good way to envision this is to think of molecules as flowing down a concentration gradient from higher concentration to lower concentration. Explain how cells control what diffuses across their membranes. Diffusion is called passive transport, and that's because it requires no cellular energy. There are 2 forms. The first is simple diffusion across the phospholipid bilayer. That's what's shown up here. There are two things that can do simple diffusion. The first are any small non-polar molecules such as oxygen, nitrogen, carbon dioxide. The second are non-polar substances such as steroid hormones or fats. Facilitated diffusion is for polar molecules and ions that can't diffuse across the phospholipid bilayer. So they require protein channels. And these are transmembrane proteins, they go all the way through, that only let specific molecules or ions pass. So over here you see one for glucose, over here you see one for amino acids, those are both polar molecules, and here you see one for an ion such as calcium. There'd be others for ions such as sodium or potassium. Compare and contrast active and passive transport. As you do, explain what powers each process. Passive transport is what we just discussed. It's when molecules or ions flow down a diffusion gradient. Here, more concentrated to less concentrated. And that's what it's all about. It's diffusion from high concentration to low concentration. It relies on the kinetic energy of the diffusing molecules or ions. It doesn't require cell energy. As you can see here, molecules are flowing down their concentration gradient. That's different from active transport, where we're pumping a molecule or ion up its concentration gradient from lower concentration to higher concentration. Over here, lower concentration to higher concentration. That requires an energy expenditure on the part of the cell. Usually that's ATP that's being converted into ADP and phosphate to power the pumping process, but also the flow of electrons can power the pumping of protons, and that's important in the creation of ATP itself. Compare and contrast endocytosis and exocytosis. These are both forms of bulk transport. In endocytosis, the membrane pinches in to surround a particle or some extracellular fluid, creating a cavity that can become a vesicle. In exocytosis, a vesicle containing something fuses with the membrane, and then that contents is dumped outside the cell. Both of these processes require energy on the part of the cell, and they both require the involvement of the cytoskeleton, which is changing the shape of the membrane. What is membrane potential? How do cells create membrane potential? How is membrane potential used? Membrane potential is an electrical charge across a membrane that creates a voltage difference. How is it created? Through cells expending energy to pump ions across their membranes. A key example, and this is a preview of a topic that we'll get into in Unit 3, is how mitochondria pump protons from the mitochondrial matrix to the intermembrane space. That gives the intermembrane space a positive charge relative to the matrix. It has all these protons inside of it. And that creates an electrochemical gradient, a voltage difference. And that pulls protons through this channel, it's called ATP synthase, back into the matrix, and that powers the creation of ATP. Some other examples, chloroplasts do pretty much exactly the same thing. And there's also a 70 millivolt charge across the membranes of nerve cells. That makes nerve impulses possible, and that's how I am able to think and teach biology, and how you are able to learn it. Is biology mind-blowing or what? I want to acknowledge how difficult and complex some of these concepts can be, and I want to encourage you to go to learn-biology.com, and with a free trial you can do the tutorials and you can use our unit reviews, and it's going to really help you to get on top of this material, setting you up for success on your unit test or the AP Bio exam. Tonicity and osmoregulation. That's the College Board's term. I wish that was just called osmosis and its consequences. So let's start by defining what osmosis is. It's the diffusion of water. Water diffuses like everything else, and that means that water is going to move from higher to lower concentration. Water is the solvent of life, so we have to pay attention to how it moves. Water flows down its diffusion gradient, just like everything else, from hypotonic to hypertonic. Those are essential terms to know. Hypotonic means relatively more water, and relatively less solute. And these terms are not absolute. They're always relative to another solution. So in this diagram, this side is hypotonic. It has a higher percentage of water molecules. It has a lower percentage of solute molecules. This side is hypertonic. And that means that it has relatively less water, a lower percentage of water molecules, and a higher percentage of solute molecules. But that's this compared to this. Now, what is that going to do? That's going to have water move down its concentration gradient, which means it'll move from here to here. After osmosis takes place, it'll look like this over here. This level will go down. This level will go up. The water actually is getting pushed up on this side. That's called osmotic pressure. It has enormous consequence. And the thing for you to memorize is that water diffuses from hypotonic to hypertonic. Here's a fabulous and fun example. A student places a gummy bear in a cup of water overnight. The next day, the gummy bear has expanded. Explain. Well, the gummy bear is mostly sugar. The water in the cup is 100% water. Water always flows from hypotonic to hypertonic. So water is going to flow from the hypotonic solution in the cup into the gummy bear. The gummy bear will expand because of osmotic pressure. Osmosis in plants. Use the principles of osmosis to explain each of the images below. On the left side, the cell is hypotonic to the environment. Water leaves the cell, the membrane peels away from the wall. That's also known as plasmolysis. The vacuole shrinks and the plant wilts. How could the environment be hypertonic to the cell? How could the cell be hypotonic to its environment? This environment has to have, for example, a lot of solutes dissolved in it so that it has a lower water concentration than is in the cytoplasm. That'll cause water to go from the hypotonic cell to the hypertonic environment. In the middle, the cell is isotonic to its environment. That's a new term. Isotonic means that the solute concentration and the water concentration are the same on both sides of the membrane. So water enters and leaves and enters at the same rate. And on the right side, the cell is hypertonic to its environment, so water flows into the cell. That osmotic pressure in the case of plants is called turgor pressure. It causes the vacuole to expand. It pushes the membrane against the cell wall. And this is a good situation for plants to be in. It makes them full, firm, happy. They're not wilted. Osmosis in animal cells. Use the principles of osmosis to explain each of the images below. Cell number 1. The cell is hypotonic to its environment. Water leaves the cell. Water always flows from hypotonic to hypertonic, so the cell shrivels up. In situation number 2, the cell is isotonic to its environment. Water enters and leaves the cell at the same rate. This is an important condition for animal cells because they have a membrane but not a wall. Cells that are kept in a tissue culture need to be kept in an isotonic solution. And in situation 3, the cell is hypertonic to its environment, and therefore water is flowing into the cells. And because there's no cell wall, the membrane can't really stop the inward force, and the cell will ultimately burst. Explain the function of the contractile vacuole in freshwater protists such as paramecia. A protist is a eukaryote that's not a plant, an animal, or a fungus. Protists in freshwater are hypertonic to their freshwater environment. That means that the cytoplasm has more solutes dissolved in it than the lake or stream that they're swimming in. And as a result, water moves into these cells by osmosis, flowing from hypotonic to hypertonic. To osmoregulate, that's a new term. Osmoregulate means to regulate your osmotic balance. The contractile vacuole, that's this organelle over here, fills with water and then contracts to expel water from the cell. If the environment becomes more hypertonic, then the cell can adapt by decreasing its rate of contractile vacuole contraction and do the reverse in more hypotonic environments. Describe the structure of leaf stomata. Stomata are pores that are on the underside of a leaf. If you took a leaf and you did a microscopic cross-section, you'd see layers of cells. The top layer and the bottom layer are responsible for waterproofing the leaf, and they're covered with wax. But nevertheless, the leaf needs to be open to its environment in order to exchange gases and other material. Each stoma, which is singular, stomata is plural, is formed by two guard cells. So here's one guard cell, here's the other guard cell. And when there's sufficient water, these guard cells buckle outward, like that, and that creates a pore that allows carbon dioxide to enter the leaf for photosynthesis, but also allows water vapor to escape. These stomata can be regulated. They can close in response to environmental cues, including water stress, and then open when water is abundant. Explain how guard cells are regulated in order to open and close the stomata. When water is available, cells adjacent to the stomata pump potassium ions into the guard cells. So we're talking about these cells and these cells, the ones that are adjacent to the guard cell, and they're represented like this over here. The pumping of potassium makes these guard cells hypertonic to the adjacent cells. Water follows by osmosis, causing the guard cells to buckle and open up. When water is scarce, the pumping stops, potassium ions flow out of the guard cells, and water follows by osmosis, causing the stomata to close down, to revert back to this form. What is water potential? Water potential is a concept that's really closely related to osmosis and osmotic pressure, but it's more quantitative. The symbol for water potential is this Greek letter psi. So water potential is a measurement of water's tendency to move from where it is to where it's not. Adding solute decreases a body of water's water potential. Adding pressure increases water potential. And water will flow from areas of higher water potential to areas of lower potential. So if you look at this device over here, it's called a U-tube, and these two parts are separated by a membrane that's permeable to water, but not to the solute. So here we are, we're adding solute to this side of the U-tube. That makes this side hypotonic, but that's another way that you can say it has higher water potential, and water is going to move from higher water potential to lower water potential. That's going to force up the level of water over here. That's going to lower it over here. Could you have explained this entire thing with just hypotonic and hypertonic? Well, I just did, but you'll see that water potential is more quantitative, and it allows for other factors to be included, such as pressure. Explain the formula for water potential. Here it is over here. In plain English, it's water potential equals solute potential plus pressure potential. So psi is water potential, psi sub s is solute potential. And remember that adding solute to water decreases its water potential. Psi p is pressure potential, and adding pressure, like if you pressed on a syringe, that increases water potential. So let's look at these two examples over here. So we've added solute to this side of the U-tube. Pressure potential is 0. There's nothing pressing down on either side, but adding solute lowered the solute potential over here to negative 0.23 megapascals, and that's the unit that's used for pressure. So the total water potential over here is minus 0.23 megapascals. The water potential over here is just 0. There's no pressure, there's no solute, so water moves from higher water potential to lower water potential. That's why it's moving from this side of the U-tube to this side. In this example, we have a potato cell or potato tissue that's been placed in water. And so again, there's no pressure on either side, it's just an open container. But the cell has a negative 1.0 megapascal solute potential, so the total water potential is negative 1.0. This is just water, so the water potential is 0 over here. Water moves from the beaker into the cell, which has a lower water potential, and that's causing water to flow into the cell. It's causing the cell to expand. All of these things related to the cell wall. In a typical lab in AP biology, you put potato tissue into water or solutions of various concentration, and you can see what happens, and you can actually quantify it based on water potential. That's what water potential is used for. You are a hero for reviewing this much of Unit 2 of AP Biology. That is phenomenal. Now what I want you to do is continue your hero's journey by going up to learn-biology.com and doing the Unit 2 review that I've set up for you. It's got flashcards, quizzes, multiple choice, FRQs, and tap challenges. You will learn so much. You'll feel so confident for your next test or for the AP Bio exam. Unit 2 is a unit for which I have a lot of great music, and I want to encourage you to check out the Cell song, my Membrane song, and my Osmosis song. If you're studying AP Bio Unit 3, Cellular Energetics, then you might be feeling nervous or anxious because AP Bio Unit 3 is a tough unit. It includes topics like cellular respiration and photosynthesis. But don't worry. The goal of this video is to teach you everything that you need to know to crush that next unit exam or the AP Bio test. Here's what we're going to cover. We're going to start with enzymes, then we'll move on to cellular energy and ATP. That'll lead us into photosynthesis, where we'll look at the big picture, the light reactions, and the Calvin cycle. We'll end with cellular respiration, again the big picture, glycolysis, the link reaction in the Krebs cycle, and we'll end with the electron transport chain. My name is Glennwolkenfeld, also known as Mr. W. I'm a retired AP Biology teacher. I love biology. To help you study, I've put together a checklist that you can download at apbiosuccess.com/checklist. I'm also the author of the learn-biology.com AP Bio curriculum and the Biomania AP Bio app. I'll tell you more about those later. Topics 3.1 to 3.3, enzymes. Describe the key properties of enzymes. Enzymes are usually proteins. There are some RNAs that act like enzymes that catalyze reactions in cells. They lower the activation energy of the reactions that they catalyze, increasing the rate of those reactions. So in this diagram, you see a reaction that's catalyzed by an enzyme, number 2, and a reaction that's not catalyzed at number 1. And the difference is that the activation energy for the enzyme catalyzed reaction is much less than the non-enzyme catalyzed reaction. Enzymes are highly specific because their active site complements the shape and charge of their substrate, which is the substance that an enzyme acts upon. Here's an active site. Here's the substrate. This is the enzyme as a whole. It would be a large protein. Here's the enzyme interacting with the substrate, and here we have the products. Enzymes are both highly specific and have a narrow set of conditions where they can function at or near their optimum. Explain. Enzymes are proteins with secondary, tertiary, and quaternary level structures that involve hydrogen bonds, ionic bonds, and hydrophobic clustering. Changing pH, temperature, or ion concentration interferes with these bonds, changing the shape of the active site, keeping the enzyme from binding with its substrate. Enzymes therefore have a pH, ionic, or temperature optimum at which the shape of their active site best fits their substrate. Environmental change can cause denaturation, a change in the shape of the enzyme that lowers or completely negates enzyme function. Describe how enzyme activity is affected by changes in the pH of its environment. Most enzymes have a pH optimum where they operate at peak efficiency. Here's the optimum right here. As pH moves above or below the optimum, enzyme performance drops. And this is the rate of enzyme activity, and you can see that as the pH drops, it goes down. As the pH increases, it goes down. Why? It's for all the reasons we talked about in the previous slide. It's that enzymes are proteins. If you change the pH, you disrupt the bonds that hold that protein in its specific shape. The result is denaturation and less good fit between the enzyme and its substrate. Describe how enzyme activity is affected by changes in the temperature of the enzyme's environment. Up to a certain point, Enzyme activity increases with temperature, and that's because there's more kinetic energy that increases molecular motion, and it increases the chance that the enzyme will bind with its substrate and therefore be able to catalyze the reaction. But at a certain temperature, beyond 2 in the graph, the enzyme will denature. It'll change its shape, reducing the enzyme's catalytic abilities because it'll no longer be able to bind with its substrate. What's the difference between reversible and irreversible enzyme denaturation? Reversible denaturation is where the restoration of optimal conditions restores the enzyme's function as it regains its optimal shape. If you can imagine that an enzyme has an optimal shape at 7, you move it up a little bit, the enzyme starts to denature, but then if you restore the pH, the enzyme shape might go back to its previous form, thereby going back to its optimal rate of efficiency. But irreversible denaturation is where the enzyme shape is permanently changed and its catalytic ability is destroyed. And this isn't exactly with enzymes, but if you think about what happens when you cook an egg, the egg white goes from clear to a solid, and it'll never go back, even once you cool it back down. That's irreversible denaturation of a protein. Imagine the same for an enzyme. Explain how enzyme activity is affected by substrate concentration. With low substrate concentrations, the probability of the enzyme meeting its substrate is low, and the product is produced at a very low rate. As substrate concentration increases, the collision and the reaction rate will also increase, but at a certain point, you get to a saturation point. And at that point, all the enzymes have their active sites interacting with substrates, so there's a peak in the rate and you don't go any higher. Compare and contrast competitive and non-competitive inhibition. In competitive inhibition, a foreign molecule, one that's not part of the cell or the organism, that's not the enzyme substrate, so this is number 4 over here, blocks the enzyme's active site. And that keeps the substrate from binding, here's the substrate, and that inhibits the rate of the reaction. It's inhibiting by competing for the active site. In non-competitive inhibition, which is shown over here, a foreign molecule, not one that's part of the organism, binds away from the active site at a region that's called the allosteric site over here. So here's the allosteric site, here's the allosteric site that's occupied by this foreign molecule. Binding at the allosteric site causes a ripple effect throughout the protein that causes a change in the shape of the active site. Therefore, the substrate can no longer bind at the active site, and that diminishes or blocks enzyme activity. Topic 3.4, cell energy. I'm Mr. W from learn-biology.com, where we believe that successful learning requires interaction and feedback, and we're so sure of that that a subscription to our website comes with a money-back guarantee. What is a metabolic pathway? A metabolic pathway is a linked series of enzyme-catalyzed chemical reactions occurring within a cell. So here you can see that these are all separate reactions, but they're linked together. Above, 1, 2, and 3 are all enzymes. A is the initial reactant. B and C are what we call intermediates, and D is the final product. Examples of metabolic reactions that you'll come to know well include glycolysis, the Krebs cycle, the Calvin cycle, all of which we'll deal with in this unit. And these reactions can be linear. So glycolysis is a linear reaction. You have a beginning point and an end point, or they can be cyclical, like the Krebs cycle and the Calvin cycle. So for example, in the Krebs cycle, this compound over here, oxaloacetate, it's the beginning compound, gets modified, and it also comes back and it's the ending compound. A similar thing happens in the Calvin cycle. What are autotrophs? Compare and contrast photoautotrophs with chemoautotrophs. Autotrophs are organisms that can produce their own food. We are not autotrophs, but plants and certain bacteria and archaea are. So photoautotrophs include plants and cyanobacteria. They use the energy and light to create organic compounds that they need to survive through photosynthesis. Whereas chemoautotrophs include some bacteria, some archaea. Archaea is that third domain. The energy for their life processes comes from a process called chemosynthesis, and that involves oxidizing inorganic substances including iron, sulfur, or hydrogen sulfide. What are heterotrophs and how do they get the energy and matter they need to live, grow, and reproduce? Heterotrophs capture the energy present in organic compounds produced by other organisms. They can be ecological consumers, decomposers, or parasites, and they get their energy and matter by metabolizing the organic compounds in organisms that they eat or absorb, or in the dead remains of other organisms. Here's a heterotroph, and here's another heterotroph. What's the difference between an exergonic reaction and an endergonic reaction? Exergonic reactions release energy and increase entropy. So here's an exergonic reaction over here, and the energy of the reactants is less than the energy of the products. And you can't see this, but like, for example, if you burn a piece of paper or a piece of wood, you start with cellulose over here, and you wind up with many unorganized atoms of carbon dioxide and water. So that's an increase in entropy because you've decreased organization. Cellular respiration, most hydrolysis reactions are exergonic reactions. Endergonic reactions require energy and decrease entropy, and examples include photosynthesis or almost any dehydration synthesis reaction. Describe the structure and function of ATP. Explain how ATP can be used to store and release energy. The structure of ATP involves a 5-carbon sugar that's called a ribose, a nitrogenous base that's called adenine, and 3 phosphate groups. The function of ATP is to power work within cells. Every cell makes its own ATP. There's no sharing of ATP between cells. And to store energy, what happens is that cells take energy from food during cellular respiration or light during photosynthesis and use that to combine ADP and a phosphate group into ATP. And to release energy for work, cells remove a phosphate group from ATP. They break off this terminal phosphate group, and that creates ADP and phosphate, and that makes energy available to do cellular work. What is energy coupling? Energy coupling is the linking of an exergonic reaction to an endergonic reaction, and that linking drives the endergonic reaction forward. Here's an example. Cellular respiration, which is exergonic, drives the formation of ATP from ADP and phosphate. So this exergonic reaction is driving this endergonic one. And example number 2 is the exergonic conversion of ADP and P is coupled to endergonic processes such as active transport. We discussed that in our Unit 2 review video. That takes energy. So ATP to ADP and phosphate makes this endergonic reaction possible. Or muscle contraction, also an endergonic reaction. It's made possible by the breakdown of ATP to ADP and phosphate. At learn-biology.com, we understand why students struggle with AP Bio. It's a hard course. The material is complex, the vocabulary is ridiculous, and the pace is withering. It's natural to feel overwhelmed and inadequate. To get an A or a 4 or a 5, you need an easier way to study, and that's why we created learn-biology.com. It has quizzes, it has flashcards, it has interactive tutorials about every topic in the AP Bio curriculum. It has a comprehensive AP Bio exam review system. Use learn-biology.com and you'll gain the skills and confidence that you'll need to ace your biology course and to crush it on the AP Bio exam. So here's your plan. Go to learn-biology.com. We've got free trials from June through March for both teachers and students. You won't believe how much you'll learn. Photosynthesis, the big picture. What happens during photosynthesis? What's its chemical equation? Is it endergonic or exergonic? In photosynthesis, using light energy, here's the sun, photoautotrophs like plants combine carbon dioxide and water to create carbohydrates. That's what the plant is made of. Oxygen is released as a waste product. It's the source of biomass and the base of almost every food chain. The formula is 6 CO2, 6 carbon dioxides, plus 6 H2O, 6 waters, with light energy to power the reaction are combined into glucose, C6H12O6, and 6 oxygens. This is an endergonic reaction for 2 reasons. It takes 2 low energy inputs, carbon dioxide and water, and converts them into a high energy product, glucose. It reduces entropy. That means it increases organization. And you can sort of count that out. So there are 12 molecules on this side of the equation, and there are 7 on this side. So we've taken something that was disorganized, made it into something more organized. A highly unorganized carbon dioxide, it's diffuse, it's a gas, and it's made into solid matter. And that's a huge decrease in entropy. When did photosynthesis first evolve? What are some of the consequences of photosynthesis? In terms of when, based on fossil and chemical evidence, about 3.5 billion years ago, that's relatively soon after the emergence of life, at 3.8 billion years ago, its consequences were vast. First of all, when the Earth first formed, there was no oxygen in the atmosphere. It's photosynthesis, which splits apart water to release oxygen, that created the oxygen-rich atmosphere, and that made our aerobic metabolism possible. And it also created an ozone layer. That's a protective layer in the atmosphere that shields us from ultraviolet radiation, and that made life on land possible. So we owe everything to photosynthesis. What are the 2 phases of photosynthesis, and what does each accomplish? We start with the light reactions. That's on this side of this diagram, and it converts light energy into chemical energy, and that chemical energy is in the form of ATP and NADPH. You already know about ATP. NADPH is like NADH. It's an electron carrier. The Calvin cycle is the second phase of photosynthesis, and it converts the chemical energy that's in NADPH and ATP into carbohydrate, and it does that by using carbon dioxide as an input, and it fixes that low energy gas into high energy sugars. Describe the role of chlorophyll in photosynthesis. Explain the absorption spectrum of chlorophyll and other pigments. Chlorophyll is the pigment that absorbs light energy in photosynthesis. And here you can see its structural formula. It's got a hydrocarbon tail that enables it to fit into the phospholipid bilayer of the thylakoids. And it's really this structure over here, this nitrogen ring with a magnesium in the center, that enables chlorophyll to help plants convert light energy into electrical energy, as we'll see in a little bit. An absorption spectrum shows the amount of light absorbed at different light wavelengths by a pigment, by a substance that absorbs light energy. And chlorophyll has 2 forms. They're different based on this functional group. Here's chlorophyll B, here's chlorophyll A, and you can see that they both absorb most energy in the blue part of the spectrum and in the red part of the spectrum, but very little in the green part of the spectrum. And that's why leaves are green, because leaves are reflecting green light, whereas they're absorbing other light wavelengths. There are other pigments that are also involved in photosynthesis. One's called a carotenoid, and they absorb other wavelengths. What is the action spectrum of photosynthesis? The action spectrum, which looks a little bit different from the absorption spectrum that we just looked at, shows how various light wavelengths drive photosynthesis. And blue and red drive the most photosynthesis, and green drives very little. This was determined by the Engelmann experiment. Thomas Engelmann in the 1800s did a cool experiment where he grew a filament of algae under light from a prism that divided the light up into its various wavelengths, and aerobic bacteria grew around the filament best in the blue and the red part of the spectrum, and they were able to do that because that's where the most oxygen was being produced. You can, in the lab, and I actually hope that you did, recreate this experiment with the famous photosynthesis spinach leaf disc experiment, where these discs of spinach leaves will rise based on the amount of oxygen that they produced. And you can set different variables like the intensity of the light or the wavelength of the light. Connect the structure of chloroplasts to the reactions of photosynthesis. So, chloroplasts, where do you find them? So here they are. This is a cross-section of a leaf. These are cells within the top part of the leaf. and these are chloroplasts. There are many per cell. There's only a couple shown here. And in terms of the structure of the chloroplast itself, it has an outer membrane and an inner membrane. The outer membrane is a vestige of the evolutionary origins of chloroplasts. There's DNA, which is a vestige of the fact that this was once an independent living cell. There's also ribosomes. They're there for the same reason. And then there are thylakoids, shown at number 5 over here. Those are membrane-bound sacs, and they contain the membrane-bound photosystems and chlorophyll for the light reactions of photosynthesis. They're organized into these stacks called grana, and surrounding them is the stroma, which is essentially the cytoplasm of the chloroplast. It contains DNA, it contains ribosomes, and it's where the Calvin cycle occurs, so that's where carbohydrates are actually created. The light reactions of photosynthesis, topic 3.5 of AP Biology. What do the light reactions produce? Where do these reactions occur? What are the inputs and the outputs? The light reactions convert the energy in light into the chemical energy of NADPH and ATP. NADPH is an electron carrier. It's like NADH in cellular respiration. ATP is the molecule that cells use to build things. It's the workhorse of the cell. Where does it occur? It occurs in the thylakoids. Oxygen is the waste product, as well as NADPH and ATP. And the inputs are light and water. The outputs of the Calvin cycle are the inputs of the light reaction. So NADP plus and ADP and P are the inputs. Those get fed into the light reactions. ATP and NADPH go out. What are the key structures involved in the light reactions? So we have a chloroplast over here, and then at N we have grana and a single thylakoid membrane. So this whole thing here is a thylakoid membrane. Within the thylakoid membrane there are photosystems. Those are complex assemblies of proteins, and they have embedded chlorophyll molecules. Those little green dots are chlorophylls. And those photosystems, those are the things that actually convert light energy, shown here at A and shown here at D, into a flow of electrons. This whole array is kind of like a solar panel that's converting light energy into electricity. They're also splitting water molecules, and that happens in photosystem 2. Let's just get this out of the way right now. In the organization of the photosystems in the thylakoid membrane, photosystem 2 comes before photosystem 1. For years, biology students have been memorizing that, and you have to memorize it too. So this is the electron pathway through which electrons flow, and at one point those electrons flow through proton pumps. They're labeled as cytochromes, but there's other stuff going on too. And what these do is they pump protons from the stroma into the thylakoid space. And over here's an enzyme called ATP synthase. And as the protons that are trapped here diffuse through this, they generate ATP. This is where NADPH is created. This is an overview. We're going to go through the details right now. Describe how the light reactions of photosynthesis create ATP. Photoexcitation of chlorophyll in photosystem 2 leads to a flow of electrons along an electron transport chain in the thylakoid membrane. That electron transport chain, that's an electrical current, and it powers a device. In this case, the device is a proton pump that's embedded in the thylakoid membrane, and that pumps protons from the stroma into the thylakoid space. So here's the stroma, here's the thylakoid space. We're pumping from the stroma into the thylakoid space, and that creates a chemiosmotic gradient. Chemiosmotic, what does that mean? Well, there are all these protons that are over here, and there are very few over here. It took energy to do that. And that gradient is a diffusion gradient, and it's also an electrical gradient. And that causes these protons to want to diffuse from the thylakoid space back to the stroma. They can't do it through any part of the thylakoid membrane except through this channel that's called ATP synthase. The ATP synthase channel is also an enzyme, and as protons diffuse through, the kinetic energy of those protons is used to power an endergonic reaction of taking ADP and phosphate and making it into ATP. Now note that there's also this water splitting complex that's part of photosystem 2. And what it does is it takes water molecules, splits them apart to create oxygen, that's a waste product, but also to create protons. And those protons accumulate in the thylakoid space. That enhances the gradient and it powers additional ATP production. Describe how the light reactions create reducing power, NADPH, that can be used in the Calvin cycle. So we're going to start over here. We're looking at photosystem 1. Photoexcitation of chlorophylls in photosystem 1 is how the process starts. That creates a flow of electrons that's flowing through the electron transport chain of photosystem 1. And over here, those electrons flow to this enzyme. It's called NADP plus reductase, and that reduces NADP plus into NADPH. Reduction, that's a thing from chemistry. Reduction is gain of electrons, so that NADP plus is going to gain electrons and hydrogen, making it into NADPH. And why? Because during the Calvin cycle, that NADPH provides the electrons and hydrogens that reduce carbon dioxide into carbohydrates. Use the Z-scheme to summarize the light reactions. So the Z-scheme is a graphical representation of everything that happens in the light reactions. This axis over here, the y-axis, shows electron energy. So what happens? Light drives electron boosting from photosystem 2. Remember, photosystem 2 comes first. And so that electron goes to a much higher energy level. And at the same time, water is split apart into protons and oxygen gas. Then there's electron flow through the electron transport chain of photosystem 2, and that goes through proton pumps that power the synthesis of ATP from ADP and phosphate. Those electrons arrive at photosystem 1. They're relatively low energy at this point. You can see that by their position on the graph. But light comes in and it stimulates chlorophylls, and another electron gets boosted to a high energy level into what's called a primary electron receptor that's in the thylakoid membrane, and that passes it off to the electron transport chain of photosystem 1. It flows to the enzyme NADP plus reductase, and NADP plus reductase creates NADPH from NADP plus and a proton. So we have the two products of the light reactions, ATP and NADPH, beautifully explained by the Z-scheme. Photosynthesis part 3, we've covered the big picture and the light reactions, and now we're going to talk about the Calvin cycle. What are the 3 phases of the Calvin cycle? So let's remember the Calvin cycle takes the products of the light reactions and carbon dioxide and uses it to create sugars. So that occurs in 3 phases. And the first phase is called the carbon fixation phase. Carbon dioxide gas is brought essentially into the biosphere. That is followed by the energy investment and harvest phase, where matter is actually pulled out, and that matter becomes part of the plant and ultimately part of you. And then finally, there's the regeneration of the starting compound. This is a cyclical reaction. This compound, RUBP, ribulose bisphosphate, is at the end and it's at the start. Now we'll go into each phase. Describe what happens during the carbon fixation phase of the Calvin cycle. The fixation phase begins as carbon dioxide is combined with RUBP. That is a reaction that's catalyzed by the enzyme Rubisco. Fun fact, that might be the most abundant protein on Earth. It creates a 6-carbon product, which isn't shown. So think about this. RUBP has 5 carbons. Each of these black dots represents a carbon atom. CO2 has 1 carbon. You'd think that would create a 6-carbon product, but immediately that 6-carbon product dissociates into 2 3-carbon molecules. So that's how we end the carbon fixation phase. Describe what happens during the energy investment and harvest phase of the Calvin cycle. we ended the carbon fixation phase with this 3-carbon molecule. And that 3-carbon product is reduced and phosphorylated. So here's a phosphorylation. In other words, ATP contributes a phosphate to this molecule. This had 1 phosphate group. This one has 2 over here. So that's a phosphorylation. And reduced. So NADPH donates an electron to this molecule. So at the end, we have this molecule G3P, glyceraldehyde 3-phosphate. It's also called PGAL, phosphoglyceraldehyde. Those are both interchangeable names. And the key thing is that this has a lot more energy than this. Why? Because it was given that energy from ATP and NADPH. This molecule can now be harvested, taken out of the Calvin cycle, and used to build plants. This is the origin of the biomass in almost any ecosystem. It's the origin of your biomass because that's ultimately where it came from. Describe what happens during the last phase of the Calvin cycle. Before we do that, I want to talk about a way to think about the entire Calvin cycle that's really going to make you think about it in a more sophisticated way. And that way pays attention to the number of carbon atoms that are present at each stage of the cycle. So we talked about how during carbon fixation, RUBP is combined with CO2. But a more proper way and correct way to think about it is that it's 3 RUBPs. RUBP is a 5-carbon compound, so that's a total of 15 carbon atoms get combined with 3 carbon dioxides. 15 plus 3 is 18. And we've talked about how the 3 6-carbon compounds immediately dissociate into 2 3-carbon compounds. So what we have over here at the end of carbon fixation is 6 3-carbon molecules. And that's a total of 18 carbons. Makes sense. 5 times 3 is 15, plus 3 is 18, and we've got 18 carbon atoms over here. Now, in the energy investment and harvest phase, during energy investment, we're just going to phosphorylate and we're going to reduce. We're adding energy, but we're not adding carbon. So what we wind up with over here is 6 molecules of G3P, glyceraldehyde 3-phosphate. 6 times 3 is 18. We had 18 over here, we have 18 over here. But what we're going to do in the harvest phase is we're going to pull one of those G3Ps out. So 18 minus 3, we're left with 15 carbon atoms, 5 times 3. Now during the next phase, these 5 G3Ps are rearranged by a variety of enzymes. That's why I have multiple arrows over here. And they're rearranged into 3 5-carbon RUBPs. And along the way, a phosphorylation occurs, and that's, again, energy from the light reactions that's invested over here. And RUBP is one of the substrates of carbon fixation along with carbon dioxide. But now we've accounted for all of our carbons. We start with these 15 over here, 15 carbons, and after the whole process is done, we again have 15 carbons. And if you can explain that, you're set up for an A and a 5 on the AP Bio exam. And that is now an appropriate time for me to say congratulations, because you've hung through one of the most difficult parts of this review. We're going to do cellular respiration next. You're a hero. Keep with it. You're going to get a 5 on the AP Bio exam. I want to acknowledge how difficult and complex some of these concepts can be, and I want to encourage you to go to learn-biology.com, and with a free trial you can do the tutorials and you can use our unit reviews, and it's going to really help you to get on top of this material, setting you up for success on your unit test or the AP Bio exam. Cellular respiration, the big picture. What's the chemical equation for cellular respiration? Is it endergonic or exergonic? Where in eukaryotic cells does it occur? The equation C6H12O6, or glucose, plus 6 oxygens yields 6 carbon dioxides, 6 water molecules, plus energy in the form of ATP. It's an exergonic reaction. It releases energy and it creates disorder. It takes an organized molecule like glucose and creates much less organization over here. And it occurs in various phases. The first phase is glycolysis. That occurs in the cytoplasm. Then the link reaction brings the product of glycolysis into the mitochondrion, and in the matrix we have the Krebs cycle. And then finally, the phase which produces the most ATP is oxidative phosphorylation, and that occurs based on enzymes and proteins along the mitochondrial membrane using the intermembrane space to create a chemiosmotic gradient. Briefly describe what happens in each phase of cellular respiration. Here we've got the whole process. Let's break it down. The process begins with glycolysis, so energy in glucose generates ATP and NADH. NADH, like NADPH that we met in photosynthesis, it's an electron carrier. And the end product is a 3-carbon molecule called pyruvic acid or pyruvate. One glucose, a 6-carbon molecule, becomes 2 pyruvates, which are each 3 carbon molecules. In the link reaction, what happens is that pyruvate enters into the mitochondrial matrix, and enzymes along the way convert it to a molecule called acetyl-CoA with 2 carbons. The carbon that's removed is released. That's 1/3 of the carbon that you exhale. And that conversion powers the reduction of an NAD plus to NADH, and that's later going to be used in the electron transport chain. In the Krebs cycle over here, what enzymes do is they oxidize these 2 carbons in acetyl-CoA, and that powers the production of 3 NADHs, 1 FADH2, that's another mobile electron carrier like NADH, and it creates 1 ATP. That releases 2 carbon dioxides, and that's the other 2/3 of the carbon dioxide that you exhale. And note that for every glucose that enters cellular respiration, the Krebs cycle runs twice, as does the link reaction. And then finally, you have the electron transport chain, the ETC, and what that does is it takes these reduced products, these reduced mobile electron carriers, and it oxidizes them, and that creates electron flow that powers through chemiosmosis the production of ATP from ADP and phosphate. That's where most of the ATP during cellular respiration is produced. Cellular respiration, parts 2 and 3, glycolysis, the link reaction, and the Krebs cycle. What happens during glycolysis include phases, inputs, and outputs in your answer. I've got a great song about glycolysis that goes like, glycolysis, come on sugar for the breakdown. It occurs in the cytoplasm. It doesn't require oxygen, it's anaerobic, and it has 3 parts to it: investment, cleavage, and energy harvest. In terms of investment, what happens is that enzymes phosphorylate glucose. So glucose is a starting compound. Enzymes aren't shown here but are implied by the arrows. Take a phosphate from ATP and plant it onto intermediate compounds. By the end of that process, you have fructose 1,6-bisphosphate. And that goes into the next phase where enzymes take fructose 1,6-bisphosphate loaded with energy and cleave it apart into 2 molecules of G3P. G3P is the same molecule that gets harvested from the Calvin cycle of photosynthesis. In the harvest phase, enzymes, again indicated by these arrows, do 2 things. They take G3P and they oxidize it. That means it's going to lose energy, lose electrons, and those electrons go to NAD+. It's a mobile electron carrier, which gets reduced to NADH, and that's later going to be used in the electron transport chain. In addition, there are other enzymes that take ADP and phosphate and phosphorylate it to ATP. They're using the chemical energy in G3P to bring that about. We're getting a gross energy yield of 4 ATPs. And that leads us to the next idea. What's the net yield of glycolysis? We get 2 ATPs. Why 2 even though there are 4 over here? It's because 2 were invested at the start of the process. So we net 2. And we also get 2 NADHs, 1 produced over here from this G3P, 1 produced over here. And we wind up with 2 molecules of pyruvate or pyruvic acid. That's still loaded with energy, and that's going to power the next phases of cellular respiration. What happens between glycolysis and the Krebs cycle? The answer is the link reaction. What we have ended glycolysis with is pyruvic acid, and that's transported across the inner and outer mitochondrial membranes. That's something that's represented by B in this diagram, into the mitochondrial matrix. As that happens, enzymes remove a CO2 from this pyruvic acid. You see this carboxyl group over here. It's basically a CO2. And that is 1/3 of the CO2 that you and any other animal that does cellular respiration, plants too for that matter, wind up releasing. Other enzymes oxidize the resulting 2-carbon molecule. So we had 3 carbons. Now we're at something with 2 carbons. It's actually called an acetyl group. And the oxidation of that acetyl group, oxidation is loss of electrons. Where do those electrons go? They go to NAD+, a mobile electron carrier. We just met it in glycolysis. And that NAD+ gets reduced to NADH, and that has electron energy that can get used in the electron transport chain. Finally, enzymes take that acetyl group and they attach it to a carrier molecule called coenzyme A, and the result is acetyl-CoA. That's the starting point for the Krebs cycle. Describe the Krebs cycle. The Krebs cycle, which I have a great song about, occurs in the mitochondrial matrix, and it's a cyclical series of reactions that generate NADH, FADH2, and ATP. It starts with enzymes, all of these are enzymes over here, enzymes taking this 2-carbon acetyl group from acetyl-CoA and transferring it from oxaloacetate, also known as oxalic acid, to citric acid, which is also known as citrate. The alternative names of the Krebs cycle are the citric acid cycle or the tricarboxylic acid cycle, TCA cycle, and that's for this carboxyl group over here, this carboxyl group over here, this carboxylic acid over here. So 3 carboxylic acids, TCA cycle. What happens next is that enzyme after enzyme oxidizes citric acid. And oxidation is loss of electrons. Those electrons get transferred to the mobile electron carriers NAD+, which becomes reduced to NADH. That happens over here, and over here, and there's also a reduction of FAD, another mobile electron carrier, to FADH2. Along the way, other enzymes over here power a substrate level phosphorylation of ADP and phosphate into ATP. There's actually a complication involving GTP, but you don't need to worry about that for an AP Bio class. For each acetyl-CoA that enters the cycle, 1 ATP, 3 NADH, and 1 FADH2 are generated. And that's probably worth memorizing. And CO2 is released as a byproduct. There's one over here. That's the other 2/3 of the CO2 that you exhale. And oxaloacetate is the starting and ending compound. That's the Krebs cycle. Cellular Respiration Part 4, the electron transport chain and oxidative phosphorylation. This is such a beautiful and important process. Let's go through it. In the previous phases of cellular respiration, glycolysis, the link reaction, Krebs, we've been creating NADH and FADH2. Those are mobile electron carriers. They've been accumulating in the mitochondrial matrix, and they diffuse over to the inner membrane where they're oxidized. Oxidation is loss of electrons. Those electrons now flow through an electron transport chain. That's this yellow arrow over here. And that's a series of membrane-embedded proteins that are in the mitochondrial inner membrane. So it's as if there's an electrical current that's flowing along the mitochondrial membrane through these various proteins. Notice that NADH comes in first. It has a little bit more energy in its electrons than FADH2, which drops its electrons off a little bit further on down the chain. Some of these electron transport proteins are proton pumps, and they pump protons from the matrix to the intermembrane space. Pumping is active transport that requires energy. Where's the energy from? It's from this flow of electrons. Notice that there's less protons over here and more over here. Again, active transport requires energy, and that creates an electrochemical gradient. There's more protons over here fewer over here. There's more positive charges over here. There's fewer positive charges over here. It's also a pH gradient because the pH is much lower in the intermembrane space than it is in the matrix. All those protons are trapped. Oxygen acts as the final electron acceptor. And what it's doing is it's so electronegative that it's pulling electrons down this electron transport chain. And as it does, it absorbs electrons and protons that are available in the matrix, and that increases the gradient. This is why you need oxygen to do aerobic respiration, because it's the final electron acceptor in the electron transport chain. Now, all of these protons have been accumulated here. They can't diffuse through any part of the inner membrane except through one channel. That's called ATP synthase. It's the same that we discussed when we discussed photosynthesis. It's a channel and it's an enzyme. As protons diffuse through, so that's diffusion, it's facilitated diffusion, their kinetic energy is used to create ATP from ADP and phosphate. That's how the electron transport chain generates ATP. Cellular respiration can be used to generate heat instead of ATP. Explain. In newborn humans and other mammals and hibernating mammals, there are cells that are called brown fat cells. They're extremely dense with mitochondria. That's where the heat is generated. When body heat is needed, hormones induce a protein channel called thermogenin, or UCP. It's also called the uncoupling channel, to form in the inner mitochondrial membrane. So here it is over here. Now notice, in normal cellular respiration, protons that are trapped have to diffuse through ATP synthase, which creates ATP. But now there's an additional channel. So protons diffuse back to the matrix from the intermembrane space without passing through ATP synthase. But all of this activity in the electron transport chain, here called the respiratory chain, still has to happen. And think about the fact that when electrons move through a wire, that generates heat through resistance. Well, basically, you can think of the electron transport chain as a wire. The electrons that are moving through it generate heat, but in this case, they generate heat without generating ATP. And that's how cellular respiration can be used to generate heat instead of ATP. How is ATP generation in mitochondria and chloroplasts similar? In Unit 3, we've talked about these two great metabolic reactions, photosynthesis and the electron transport chain of cellular respiration. And there are deep similarities to the way that they work. And this kind of cross-topic thinking is essential to your success in the AP Bio exam. Both of these processes use an electron transport chain to pump protons to an enclosed space, creating a proton gradient. And in photosynthesis, here's the electron transport chain. We're pumping protons from the stroma to the thylakoid space. In cellular respiration, we're pumping protons from the matrix to the intermembrane space. And both use a subsequent process of chemiosmosis, diffusion of protons through an ATP synthase channel to generate ATP. And that's not a coincidence. As we'll see in Unit 7, where we'll talk about evolution, the similarities that are present between mitochondria and chloroplasts indicate that at some point in very ancient history, they had a common ancestor. ATP synthase evolved once, and then it became shared by the ancestors of chloroplasts and mitochondria. It's an incredible ride, seeing biology and seeing evolution in process. Cellular respiration, part 5, anaerobic respiration and fermentation. Compare aerobic and anaerobic respiration. Aerobic respiration, oxygen is required. It involves essentially the whole shebang of respiration that we've talked about, glycolysis, plus the link reaction, plus the Krebs cycle, plus the electron transport chain, generate a lot of ATP, 32 ATPs approximately for every molecule of glucose that enters the cycle. Most of the ATP is generated over here in the mitochondria. There's a small amount that's generated through glycolysis. Anaerobic respiration occurs when oxygen is lacking or insufficient, or I should add, when the organism doesn't have the enzymes to do aerobic respiration. Glycolysis is really the key part of anaerobic respiration. So it involves glycolysis followed by fermentation, and it generates a total of 2 ATPs, and it occurs entirely in the cytoplasm. The mitochondria are not involved. What is fermentation and why does it occur? Fermentation is glycolysis that's followed by reactions that regenerate NAD+. So here we have one form of fermentation. It's called alcohol fermentation. Here we have lactic acid fermentation. But the key thing is that they produce NAD+. That's kind of the opposite of what glycolysis does. Why does fermentation happen? It's because if you're an aerobic organism, but you need to keep on moving and there's not enough oxygen available because you're working so hard, then you can continue to get 2 ATPs per every glucose. And that sure isn't as good as 32 or more that you get from aerobic respiration, but at that moment you just can't do it. Glycolysis, however, can only create those 2 ATPs if NAD+ is available. It's a substrate for one of the reactions of glycolysis. So 2 ATP are better than none, and that's why animals like us that do lactic acid fermentation perform it when oxygen is unavailable. Compare and contrast alcohol and lactic acid fermentation. Alcohol fermentation, really what we're talking about is ethanol fermentation, because there are other alcohol fermentations, occurs in yeast. When you're baking bread and you use yeast, you're doing an alcohol fermentation. Enzymes remove a carbon dioxide from pyruvic acid. Remember, that's the molecule that ends glycolysis. That produces acetaldehyde, and acetaldehyde is then by other enzymes reduced to ethanol. Well, reduction is gain of electrons. Something has to lose electrons, and NADH is oxidized to NAD+. That allows glycolysis to continue. Interesting and fun fact, this CO2 that makes up the bubbles in beer, which come about through an alcohol fermentation, that also makes up the bubbles that cause bread to be spongy and have its spongy form. Now, lactic acid fermentation occurs in muscle tissue under anaerobic conditions. It's a little bit simpler than alcohol fermentation. What happens is that pyruvate is reduced to lactic acid, and again, as that reduction occurs, something has to be oxidized, and NADH is oxidized to NAD+. That allows glycolysis to continue. There are all kinds of anaerobic sports like weightlifting or doing push-ups or things like that, and you're doing them at a level where muscle tissue can't really get enough oxygen. So during that time, they're doing lactic acid fermentation. You feel that lactic acid build up, and then eventually you have to stop exercising until you can recover aerobically. Oh my goodness, I can't tell you how much I admire you for hanging on until this point and learning so much biology. Science Music Videos has so much music related to AP Bio Unit 3. I'll put the links below. Have a big party, sing these songs. It's a great way to learn this material. If you really want to crush your AP Bio course and the AP Bio exam, then sign up for that free trial at learn-biology.com. Our AP Bio reviews, our interactive tutorials, our quizzes, our flashcards, our games are going to help you get to the level that you want to get to for your AP Bio success. If you're feeling overwhelmed and exhausted by the intensity of AP Bio as you prepare for the AP Bio exam or a Unit 4 test, don't worry, you've come to the right place. In this video, we'll be reviewing all of Unit 4. That includes 1. Cell communication, cell signaling, and signal transduction. 2. Feedback and homeostasis. 3. Cell division and the cell cycle. And 4. Cell cycle regulation, cancer, and apoptosis. I'm Glenn Wolkenfeld, also known as Mr. W, and I love teaching biology to help you study. I've put together a checklist that you can download at apbiosuccess.com/checklist. Topics 4.1 to 4.4, Part 1, Cell Signaling, the Big Picture. Cells are constantly communicating with one another. It's a basic feature of life. Cells are never really on their own. They're always in populations or they're in multicellular organisms. So there's direct cell-to-cell communication that you see here, where there's some kind of junction between two adjacent cells, and molecules can pass between those two cells, and that enables one cell to change the behavior of the other. There's also communication that happens through signals. So here you have a cell that's producing a molecule. It's secreting that molecule into the bloodstream or into the extracellular fluid, and that message is going to be picked up by a target cell. These signals are known as ligands, and there are basically two kinds. If the ligand, the signal, is traveling a long distance through the bloodstream from a gland to another part of the body, that's called a hormone. When cells are in relatively close proximity to one another, they produce local regulators, and that's for short distance cell-to-cell communication. Describe direct contact communication in plant and animal cells. Plant cells can be connected by plasmodesmata. These are bridges involving the cell wall and the membrane. These bridges allow signaling molecules, water, and other substances to diffuse from cell to cell. Animal cells have gap junctions. These are built of protein channels that connect adjacent cells. They allow a variety of substances, including signaling molecules, to move from the cytoplasm of one cell to the next. What are ligands? Ligands are signaling molecules. Many ligands are hormones, and we'll discuss some in depth in this review. What they do is they bind with receptors based on complementary shape. And here I've represented that very simply with a kind of circle and an arc. But because this is biology, you know that the shapes will be extraordinarily complex, like enzymes and substrates. Binding leads to a cellular response, and the mechanism by which that happens is going to be most of what we're going to talk about in this video. What is quorum sensing? This is a kind of cell communication that's seen in biofilm formation in bacteria, and if this seems out of context, these are films that can form, for example, on your teeth, leading to the buildup of plaque. What happens is that bacteria, so here's a single cell, be like one of these over here, what they do is they release signaling molecules, shown over here at number 2, and those bind to cytoplasmic receptors. So those are receptors that are actually inside the cell. They're not on the membrane surface. When the signal exceeds a certain intensity, so when there's a lot of cells around, that's a quorum. And those signals will be binding with the receptors, and that will activate genes. And that gene activation will lead to the expression of, for example, in this case, it looks like what these cells are doing is they're producing a biofilm, a polysaccharide that forms that biofilm. What's the takeaway? Twofold. The first is that all cells communicate, even bacteria, and the second is that you should brush your teeth so that you don't get a buildup of biofilm leading to plaque. Cell signaling involves 3 phases. List them. The 3 phases are reception of a ligand, that's what's happening over here. There's signal transduction, where the initial message gets changed into another kind of message that can go into the cytoplasm. That often involves amplification of the signal. And finally, there's a cellular response. We're now going to expand on the material in the previous slide. What happens during the reception phase of cell signaling? The signal molecule, also called a ligand, as we've talked about before, binds with a receptor molecule. Here you can see a receptor that has a more realistic depiction, and you can see how it's embedded in the phospholipid bilayer of the cell membrane. That binding is based on complementary shape. What happens during the transduction and response phases of cell signaling? The receptor during the transduction phase interacts with membrane proteins to produce a second messenger. So something will be happening between here and other proteins in the membrane to bring about this second messenger. The second messenger with other relay molecules will bring that message to the cytoplasm, activating enzymes, or the nucleus, where we'll have the activation of genes. How is the mechanism of steroid, nonpolar hormones different from that of water-soluble hormones? The hormones we've talked about so far, the cell communication mechanisms we've talked about, have all involved water-soluble hormones. But what if the hormone is nonpolar, a steroid hormone like estrogen or testosterone? In that case, These hormones, which are non-polar, can diffuse through the phospholipid bilayer. And once they're there, they can bind with a cytoplasmic receptor. That forms a receptor hormone complex, and that is capable of diffusing into the nucleus, where it acts as a transcription factor, something we'll talk about in AP Bio Unit 6. But all you need to know for now is that that can activate genes. So the DNA gets made into RNA. The RNA goes into the cytoplasm. It gets read by a ribosome and gets made into a protein. Water-soluble hormones, they're capable of binding with receptors, they interact with second messengers, and they bring about a cellular response. And in general, these responses are slower, but more longer lasting. These responses are quicker. Topics 4.1 to 4.4, Part 2. Now that we've had an overview of cell communication, we're going to look in depth at an illustrative example, epinephrine and G protein coupled receptor systems. Before we delve into this, I want to let you know that I have a song that's a rap about cell communication, which covers much of this material.
Cellular communication works through phases 3.
The context for what we're about to review or learn is the fight or flight response, and that has effects throughout our bodies. One of the effects is the effect on the adrenal glands, which produce a hormone that's called epinephrine or adrenaline, and that acts upon the liver to get it to produce glucose that goes into the blood as part of the fight or flight response. Epinephrine is also known as adrenaline. It's a polar water-soluble hormone. You can see these hydroxyl groups over here and over here. Here's an amino group. This is not going to be able to diffuse into the cytoplasm. It's going to bind at the membrane. Note that epinephrine's effects are widespread but tissue specific. So epinephrine is going to get released from the adrenal glands into the bloodstream. It's going to go everywhere. It's going to touch every cell in your body, but only tissues with receptors are going to respond. That response will differ based on tissue types. So all of these are adaptations that are part of the fight or flight response. So over here we'll decrease digestion, because when you're trying to fight off some mortal threat, you don't need to be digesting at that moment. You want to increase your heart rate so that you can deliver more food and oxygen to your cells. Pupil dilation, more light, you're going to have better senses. Conversion of glycogen to glucose, that gives your cells, your muscle tissue, more energy to fight or flee. And bronchial dilation allows you to get more oxygen into your lungs so you can deliver more oxygen to the cells of the body. Epinephrine interacts with cells in the liver, and it induces changes that causes those liver cells to take stored glycogen, that's a polysaccharide, and to hydrolyze it into the monomers of polysaccharide, glucose. That glucose then diffuses into the bloodstream, and there it goes to the muscles of the body and to other organs as well, and that provides energy to fight or flee as part of the fight or flight response. The question for us is, how does epinephrine get liver cells to bring about this response? So we're now looking at the off state before epinephrine is released, and in this moment the receptor is unbound. There's no epinephrine in the system. There's a nearby membrane protein that's called a G protein. A G protein is not a receptor, it's a membrane embedded protein, and it can oscillate between two states. Now it's off, it's inactive. Nearby the G protein is a membrane embedded enzyme, not a receptor, but an enzyme that's called adenylyl cyclase. That is actually the correct pronunciation, and it's also in the off state. And as a result, it's not activating the second messenger. What happens when epinephrine enters the system? First thing that happens is that epinephrine binds with a G protein coupled receptor. So here's epinephrine, and here it's binding with the receptor. This is a complicated protein, and we've talked about allosteric shifts in relationship to enzymes, where when something binds at an allosteric site, it can then change the active site. Well, the same mechanism is at work here. Epinephrine's binding over here, and that change kind of ripples through this protein, and it induces a change over here. Now, right at this moment, the nearby G protein is still dormant. It's still bound to GDP. GDP is a relative of ADP, and it's the low energy form. It can oscillate between this low energy form and a high energy form that we're going to see in a minute, and that happens when the G protein becomes activated. What's the effect on the G protein of epinephrine binding with the receptor? Well, the G protein is then able to interact with the receptor. We noted before that the receptor has changed on its cytoplasmic side, and that enables the G protein to interact with that part of the receptor, and that causes the G protein to discharge GDP, that's the low energy form, and to bind with GTP, that's the high energy form. And again, notice that this has 3 phosphates over here, just like ATP. This only has 2 phosphates over here, like ADP. The result is that the G protein now becomes activated. So what happens to the G protein once it's bound with GTP? It drifts in the membrane. It ultimately binds with adenylyl cyclase, this membrane-embedded enzyme. That activates adenylyl cyclase. And adenylyl cyclase's substrate is ATP, and it converts it into a molecule called cyclic AMP, which is the second messenger in these G protein coupled receptor systems. Note that ATP is triply phosphorylated. This only has 1 phosphate, and the AMP stands for adenosine mono as in 1 phosphate. What have we done? We've taken our initial messenger and we've transduced it, creating the second messenger. So let's review what we've talked about so far. We've talked about reception. We have the ligand, which is epinephrine. It binds with the G protein coupled receptor. The receptor changes shape on its cytoplasmic side. It interacts with the G protein, causing it to discharge GDP, which is what it's bound to when it's dormant, and bind with GTP, which is what it binds with when it's active. The G protein then in turn can activate adenylyl cyclase, which takes its substrate, ATP, and converts it into cyclic AMP, the second messenger. What we're going to look at next is the cellular response. The second messenger, cyclic AMP, is going to activate a chain of relay molecules. These are called kinases or kinases. And this activation involves one kinase activating the next kinase, activating the next. I've only put 3 in this chain, but there can be many, many more, and that's called a phosphorylation cascade. How does that phosphorylation cascade work? The kinases are activated by phosphorylation, by gaining a phosphate. And once they're activated, what they do is they activate the next kinase in the chain. So here we have protein kinase 1 that acts upon protein kinase 2 by phosphorylating it. So now protein kinase 2 is active, phosphorylated. What does it do? It activates protein kinase 3 by phosphorylating it. I've only shown 3, but there can be many more in this chain, and we get this domino-like effect of one kinase activating the next and activating the next. Once we get to adenylyl cyclase, then each step involves multiple activations. Adenylyl cyclase will activate many cyclic AMPs. Each of these cyclic AMPs will start different phosphorylation chains, and the result is signal amplification. We had 1 epinephrine enter the system, but by the end, and I couldn't of course depict that here, but you'll have the activation of millions of enzymes to bring about a massive cellular response. In the case of liver cells and the way that they're acted upon by epinephrine, the response is activation of the terminal enzyme, which is glycogen phosphorylase. And what glycogen phosphorylase does is it converts glycogen, again a polysaccharide, into glucose, a monosaccharide. That glucose diffuses into the blood, giving you energy for the fight or flight response. We've seen how epinephrine, working through this G protein coupled receptor system, can mobilize this massive cellular response. This is incredibly quick, and it has to be because it's part of the fight or flight response. But after the threat has been escaped from or dealt with, the system also needs to be shut down, and that needs to happen quickly as well. To explain that, I'm going to show you this excerpt from my cell communication wrap. Enjoy.
Back to the ligand of the receptor. It only stays for a moment before it diffuses away. So when the thread is gone, the cascade gets shut down. With no adrenal secretion, the receptor's unbound. G protein drops the phosphate and bound to GDP. It goes back to sleep, stops its activity. Bound to GDP, it no longer stimulates adenylyl cyclase, which no longer creates cyclic AMP, so the second message stops. Kinase phosphorylation quickly drops as other enzymes, protein phosphatases, clip off phosphates, turn off kinases. Glycogen phosphorylase stops hydrolyzing glycogen, so blood glucose normalizes. Liver cells return to their resting state. I love how G proteins let cells communicate.
I want to acknowledge how difficult and complex some of these concepts can be, and I want to encourage you to go to learn-biology.com, and with a free trial you can do the tutorials and you can use our unit reviews, and it's going to really help you to get on top of this material, setting you up for success on your unit test or the AP Bio exam. Topic 4.5, feedback and homeostasis. What is homeostasis? What are feedback mechanisms and how do they connect to homeostasis? Homeostasis is the tendency of a living system to maintain its internal conditions at a relatively constant, optimal level. Your body temperature, for example, stays around 37 degrees Celsius, 98.6 Fahrenheit, despite fluctuations in the external temperature. The changes in your body to maintain that temperature, that's a great example of homeostasis. Feedback is when the output of a system is also an input. So here we have some system, here's the thing that's coming out, and feedback is when it feeds back into the system. So the output is also an input. And feedback can do two things. One is connected to homeostasis. It can allow organisms to maintain homeostasis as they respond to internal and external changes, just as I told you with temperature, or it can accelerate internal changes and drive a process towards a conclusion. This is generally connected with what's called negative feedback, and this is connected with what's called positive feedback. Let's talk about set points. What is a set point? How are set points used in negative feedback? First, a word about the method here. We're going to talk about this in relationship to your home heating and cooling system. It's much easier to understand this before we look at the biological examples. So a set point is where you set the thermostat. It's the value around which a homeostatic process fluctuates. So above, the set point for the thermostat is 68 degrees Fahrenheit. In negative feedback, the output of the system feeds back to the system in a way that decreases the system's output. So here we have the set point. Here's a measurement of the temperature. The set point is above the temperature. That'll send a signal to turn on the furnace. That'll generate heat. Heat feeds back to the system. There's a thermostat over here with a thermometer. That will increase the temperature, and then when we get to 68 degrees Fahrenheit, the set point, the system will turn off. Negative feedback promotes homeostasis, returning a system to its set point. How can antagonistic negative feedback loops be paired to promote homeostasis? So, same goal, keep the temperature at 68 degrees. Well, here we have a negative feedback system that responds when conditions are above the set point. So, temperature 70, the set point is 68. What will you do? You'll have a signal that turns on an air conditioner. That will put out cool, it's a real word, and that will feed back to the system, and eventually it'll get the system to turn itself off. There's been cooling, and we've therefore lowered the temperature. But we still have a heating system, and if the temperature goes below the set point, then that system would turn on, it would release heat. That would feed back to the system, and that would wind up turning the system off. So we have paired antagonistic systems, one for cooling, one for heating. Now we're going to look at some biology. Let's talk about how feedback works in blood sugar homeostasis. It's important to your body to maintain your blood sugar levels, your blood glucose levels. The main hormone that controls that is insulin, and it's a negative feedback system. In response to high blood glucose levels after eating a sugary or a starchy meal, your pancreas will release the hormone insulin, just represented here as a triangle. In the liver, insulin will bind at a receptor over here, and that will unleash a signaling cascade, and that talks to a glucose transporter. That's a channel that allows for facilitated diffusion. If glucose is in higher concentration outside than inside the cell, it'll diffuse into liver cells, and the glucose will get converted into glycogen, which is a storage polysaccharide, or it'll get converted into fat. Blood glucose levels down, homeostasis restored. But as with our home heating and cooling system, insulin is paired with another hormone. So let's explain how insulin and glucagon maintain blood glucose homeostasis. Our blood glucose has a set point about 90 milligrams per deciliter. Above the set point, the pancreas will release insulin. Eat a big meal, insulin gets released, and your liver, fat, and muscle cells, which aren't shown, will absorb glucose and store it away as glycogen. Below the set point, that's when you've gone a little while without eating, what will happen is that the pancreas will release a second hormone called glucagon, and that will induce the liver to convert its stores of glycogen into glucose. Glucose goes into the blood, homeostasis is restored. An important AP Bio skill is explaining what happens when systems get disrupted. And here we're going to explain how blood glucose homeostasis breaks down in type 2 diabetes. That's also called adult onset diabetes, but increasingly it's happening in teens and even children due to the obesity epidemic in the United States. We're going to look at normal metabolism first, which we just discussed. Here we see insulin, in response to a high blood glucose level, is binding with the insulin receptor. There's a signaling cascade that causes the glucose channel to open. There's glucose absorption into the cells. Blood glucose level falls. Homeostasis restored. In people with type 2 diabetes, what happens is that the cells become insulin resistant. There's glucose in the blood. There's insulin released from the pancreas, but the binding of insulin does not lead to the signaling cascade. And because of that, the glucose channel remains closed. Because of that, blood glucose level stays high, and that damages organs and tissues. Breakdown of homeostasis leading to type 2 diabetes. We just discussed adult onset type 2 diabetes. How does that compare with type 1 diabetes? Type 1 is also known as juvenile diabetes. It's an autoimmune disorder, and basically what happens is that cells of your immune system attack the pancreas cells that produce insulin. Therefore, those cells can no longer secrete insulin, and therefore, even in response to a high blood glucose level, insulin is not secreted. That can only be treated by insulin injections. Type 2 diabetes we've just discussed, also called adult onset, and that involves insulin resistance where the receptor is become insensitive to the insulin signal. All of the biological feedback loops that we've talked about so far relating to blood sugar have involved negative feedback loops. What about positive feedback loops? How does positive feedback work? Explain how positive feedback works during childbirth. So in positive feedback, the output of a system feeds back into the system, increasing the system's activity and output. It doesn't involve quieting that leads to homeostasis. It involves acceleration. It drives a biological process such as childbirth to a conclusion, after which the system shuts down. In childbirth, the growth of a baby activates uterine stretch receptors. The uterus, among other things, is a big muscle. When that muscle is stretched out, it sends messages to the brain. The brain releases oxytocin, a hormone. That oxytocin then circulates in the blood. When it's picked up by receptors in uterine cells, that leads to more contraction. That increased contraction leads to more oxytocin release, and ultimately that continues until the baby is born. Explain how feedback leads to fruit ripening. If you were to pause the video and take a look at this diagram, I'll bet you that you could figure it out. This is a positive feedback system. Ripening in fruit leads to the release of a hormone that is a gas. It's called ethylene. Ethylene receptors in nearby fruit pick up the ethylene, and that induces additional ripening and more ethylene production. That increased concentration of ethylene accelerates the ripening process in all the fruit, leading to more ethylene. So eventually all the fruit ripens. In fruit shipping, you can prevent that by pumping carbon dioxide into the storage container where the fruit are, and that suppresses the ethylene, and that's how you can ship fruit long distances. At learn-biology.com, we understand why students struggle with AP Bio. It's a hard course. The material is complex, the vocabulary is ridiculous, and the pace is withering. It's natural to feel overwhelmed and inadequate. To get an A or a 4 or a 5, you need an easier way to study, and that's why we created learn-biology.com. It has quizzes, it has flashcards, it has interactive tutorials about every topic in the AP Bio curriculum. It has a comprehensive AP Bio exam review system. Use learn-biology.com and you'll gain the skills and confidence that you'll need to ace your biology course and to crush it on the AP Bio exam. So here's your plan. Go to learn-biology.com. We've got free trials from June through March for both teachers and students. You won't believe how much you'll learn. Topic 4.6, the cell cycle. On a big picture level, what does mitosis do? List 3 of its key functions in living things. Note for this question that mitosis, as it often is synonymous with eukaryotic cell division. Mitosis duplicates the chromosomes of a eukaryotic cell, transmitting that cell's entire genome to its daughter cell. So here you have the parent cell. It's got 2 chromosomes. Here those chromosomes have been duplicated. Now they're being pulled apart. And now you have daughter cells, each with 2 chromosomes. And each one is going to be an exact clone of its parent cell. In a multicellular organism like you and me, mitosis is how an organism grows and repairs itself. Remember that you started life, as all multicellular beings do, as a single cell. In a unicellular eukaryote, like a paramecium or an amoeba, mitosis is how reproduction occurs. Describe what happens during the cell cycle. The cell cycle, as you can see in this diagram, can be divided into 2 main phases. So the outside orange part of the circle, that's interphase. This yellow part over here, that's mitosis or M phase, represented by the letter M. During interphase, you can subdivide 3 basic phases, but the cell doesn't appear to be dividing during interphase. The first is G1, and during G1, the cell increases in size. G1 stands for growth phase 1. During S, which stands for synthesis, you have DNA replication or chromosome duplication. And during G2, growth phase 2, you have the growth of the structures that are required for cell division. During M phase, you have mitosis, separation of the chromosomes followed by cytokinesis. So at the end, you have 2 daughter cells that are clones of the parent cell. Describe the phases of mitosis. We begin with interphase, during which the cell grows and replicates its DNA. During that time, it doesn't look as if it's dividing, but DNA replication, for example, has occurred. During prophase, chromosomes, which are spread out as what's called chromatin during interphase, they condense into these X-like structures. The nuclear membrane disintegrates, and a spindle apparatus, which is these fibers over here, you can see the entire apparatus in the next phase, start to grow from each centrosome. During metaphase, the spindle fibers grab onto the chromosomes and they pull and push them to the cell equator. If you can sort of imagine a line running down from top to bottom, that would be the cell equator. I talked about this X-like formation that the chromosomes are in. That's because each chromosome is doubled and consists of 2 clones called sister chromatids. During anaphase, the spindle pulls the sister chromatids apart and starts dragging them to the opposite ends of the cell. At the same time, there are what are called non-kinetochore microtubules. A kinetochore is like a handle on the chromosomes that these spindle fibers use to pull the chromosomes apart. But there are other fibers that basically push on one another, and that causes the cell to elongate. During telophase, a new nuclear membrane starts to grow around each set of chromosomes. The chromosomes spread out into their interphase formation so you can't really see them anymore. And a nucleolus reappears in each cell. It disappeared during interphase. The nucleolus makes ribosomes, and ribosome production shuts down during most of mitosis. Finally, during cytokinesis, the cell splits apart into two daughter cells. I have a great song about mitosis, and I'll put the link below.
Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew.
Explain the importance of the G0 phase of the cell cycle. Basic idea here is that not all cells go through the entire cell cycle. So if you have highly specialized cells like a nerve cell or a muscle cell, they'll leave the cell cycle and they'll enter into G0, after which they won't divide. Certain stimuli, however, can induce cells in G0 to re-enter the cell cycle. But for the most part, the nerve cells that you have are the nerve cells that you're going to have, which is why a traumatic spinal cord injury is something that you can't recover from. Topic 4.7, regulation of the cell cycle. I'm Mr. W from learn-biology.com, where we believe that interaction and feedback is what leads to deep, substantial learning. We're so sure of that we provide a money-back guarantee that comes with your subscription. Describe the role that checkpoints play in regulating the cell cycle. Cell cycle checkpoints are moments when the cell checks its internal conditions and decides whether to progress to the next phase of the cell cycle. If certain molecules are in the right concentration, then the cell continues through the cell cycle, and if not, the cell moves into G0 or might initiate what's called apoptosis. We'll explain that in a moment, which is called programmed cell death. The primary checkpoints to know about for AP Bio are the G1 checkpoint over here, the G2 checkpoint, and the M checkpoint. What is apoptosis? I've talked about that several times. Note that the second P is silent. Apoptosis is programmed cell death. It's part of a signaling cascade that involves the mitochondria and the nucleus. It's highly regulated, which is very different from cell death that results from traumatic cell injury. Cells are broken down into cytoplasmic fragments that are called blebs. You can see some of those over here and over here. And blebs are consumed by cells of the immune system, preventing cellular debris and enzymes from damaging nearby cells. What are cyclins and cyclin-dependent kinases? Cyclins and cyclin-dependent kinases are important internal regulators of the cell cycle. The cell cycle is regulated by outside signals, but also by internal conditions. Cyclins are molecules whose concentration rises and falls throughout the cell cycle. So like, for example, you can see that cyclin E rises right before the S phase. Cyclin A rises right before G2. The cell has mechanisms to read the level of these cyclin concentrations. Kinases, which we discussed previously in the context of cell communication, those are molecules that activate other molecules, often by phosphorylating them. They're not shown. And cyclin-dependent kinases, or CDKs, are kinases that respond to rising and falling levels of cyclin levels. Now we're going to put that all together and see what some of the mechanisms are that regulate cell division. Explain how the interactions between cyclins and cyclin-dependent kinases control the cell cycle. So CDKs, here they are over here, are present at a constant level throughout the cell cycle. By contrast, the level of cyclins that we saw in the previous slide rise and fall. When cyclin levels are high, the cyclins bind with CDKs to form a complex called maturation promoting factor, but a good way to remember that is just think of it as mitosis promoting factor. Because once you have MPF, that allows the cell to pass through the G2 checkpoint and actually divide. During M phase, however, the cyclin is broken down, and that allows the process to repeat in each daughter cell when it grows to the appropriate size and gets ready for cell division. We talked about the cell cycle. Now let's talk about dysregulation of the cell cycle. What's the connection between cell division and cancer? What are the two types of genetic mutations that are connected to cancer? Cancer is a disease of unregulated cell division. As opposed to cells staying in place doing what they're supposed to do, they become rogue players pursuing their own destiny at the cost of the organism. Mutations in genes that are called proto-oncogenes increase cell division by creating too many growth factors. Growth factors are things that stimulate cell division within a cell itself or within other cells. And there are other kinds of genes that are called tumor suppressor genes, and what they do is they remove cell division inhibitors, the kind of checkpoints that we've seen in previous slides. So in normal cells you have the brakes, those are tumor suppressors, and you have the accelerator. Those are growth factors, but they act at appropriate times. When cells become cancerous, it can be for one of two or actually both reasons. You can have mutated tumor suppressors that can't prevent cell division even when cell division shouldn't be happening. And you have growth factors that promote cell division at unneeded moments. Describe how a mutation in the ROS proto-oncogene can induce a non-cancerous cell to become cancerous. What we're going to do now is really cool because we're going to connect what we've learned about the cell cycle and its control to what we learned about previously relating to cell communication. So ROS is a G protein over here, and as a proto-oncogene, it only becomes active when an outside growth signal binds with ROS's coupled receptors. So here we have binding that activates ROS, it picks up GTP, that sets off a phosphorylation cascade, and that leads to cell division. So this is in a healthy, normal system. ROS only gets activated when there's a ligand. When ROS becomes cancerous, when it mutates from a proto-oncogene to an oncogene, then it becomes constitutively active. And constitutively means it's part of its nature to be active. Normal ROS is only active, it only binds GTP when the receptor binds a ligand. But this ROS, which is an oncogene, is constitutively active. It's binding GTP even in the absence of a growth signal, and because it's always active, the growth factor over here is overproduced, and that results in too much cell division. This is connected with about 30% of human cancers. Describe how a mutation in a tumor suppressor gene such as p53 can contribute to the development of cancer. p53 is a tumor suppressor gene. When cells experience DNA damage, a signaling cascade activates p53. So here we have DNA damage, it's detected, there's a signaling cascade, and now p53 is activated. If the damage can be repaired, then what p53 will do is it'll halt the cell cycle while DNA repair enzymes fix the damage. So we're going to fix up the DNA, that's what's happening over here. If the damage is too great, then p53 will initiate a whole signaling cascade that'll cause the cell to initiate apoptosis. So either we have repaired DNA that occurs while the cell cycle is halted, or the cell will self-destruct. Cancer's been prevented. If mutations lead p53 to become non-functional, then the cell will continue to divide even with damaged DNA. So here we have DNA damage, it's been detected, but p53 can't do anything about it. Therefore, there's no stop signal. The cell will continue to divide, and that'll increase the chance of the cell acquiring further mutations that can lead it to become cancerous. Again, dysregulation of a signaling cascade, dysregulation of a cell cycle repair mechanism leading to cancer. If you're studying for the AP Bio exam or for a Unit 5 AP Bio test, and you're feeling nervous and scared about topics like meiosis and dihybrid crosses or linkage and recombination, that makes so much sense. Those are difficult topics, but don't worry. In this video, I'm going to teach you everything that you need to know so that you can crush that next test or the AP Bio exam. My name is glennwolkenfeld, also known as Mr. W. I'm a retired AP Biology teacher. I love biology. Here's what we're going to cover in this video. We're going to start with meiosis. That'll lead us to sex determination. And that'll lead us to an investigation of what can go wrong in meiosis. Then Mendelian genetics. We'll start with key concepts. We'll look at monohybrid crosses and dihybrid crosses. Then non-Mendelian genetics, which includes linkage and recombination, sex-linked alleles, non-nuclear inheritance, and genotype-environment interaction. To help you study, I've put together a checklist that you can download at apbiosuccess.com/checklist. Meiosis, the big picture. What is meiosis? Why is it important? Meiosis is how sexually reproducing eukaryotes, that includes animals, plants, fungi, protists, transmit genes from one generation to the next. It creates variation between parents and their offspring, and it creates variation among the offspring. Describe the life cycle of sexually reproducing eukaryotes. In eukaryotes, adults have specialized tissues, testes and ovaries, for creating gametes. Gametes are sperm and egg cells, and they do that through this process that we'll be exploring called meiosis. The sperm fertilizes the egg, and that produces a zygote, or a fertilized egg. That zygote then divides and develops, the tissues differentiate to produce an adult organism. In relation to meiosis, compare haploid and diploid cells. These are super important terms for understanding meiosis. Parents have two sets of chromosomes in all of their body cells, with the exception of their gametes. Those chromosomes are paired. So like, for example, here's chromosome 1. There are two of them. Here's chromosome 2. There are two of them. One was inherited from one parent, one was inherited from the other. Those pairs are said to be homologous, and that's a term that we'll explain in the next slide. When parents pass on their chromosomes to the next generation, they can't pass all of them on. If they did, then the chromosome number would double in every generation. So what happens in meiosis is a halving of the number of chromosomes. And the half number of chromosomes kind of rhymes with the word half, or it begins with the same prefix. It's called haploid. So notice that there are 4 chromosomes down here. There are only 2 over here. What's the difference? The difference is what happened during meiosis, which is division of cells that involves reduction. Reduction division. Define homologous chromosomes. Homologous chromosomes are the matching chromosomes that you inherited from your parents. So, for example, here's chromosome 3. One of them came from your mom, one of them came from your dad. Here's chromosome 4, it's the same thing. Here's chromosome 5, all the way on down the line. They are not identical. How could they be? Your mom and dad aren't identical, so the chromosomes that they passed on to you wouldn't be identical. They do have the same genes in the same order, but the alleles, the specific code that is in the location where those genes are found, that might be different. Let's use the analogy of a gene as a recipe. Well, the one that you inherited from your mom, if that were a recipe for tomato sauce, maybe that one has a lot more garlic, and the one that you inherited from your dad, that might have a lot more basil in it. Well, let's now think more biologically. If C refers to a specific protein, then the DNA that's coding for a specific sequence of amino acids might be slightly different in what you inherit from your mom and your dad. And that might even be to the extent where the amino acid sequence of that protein differs. So the genes are the same, but the alleles might be different. That's what homologous means. More essential meiosis genetics vocabulary: compare and contrast germ cells, gametes, and somatic cells. Germ cells are the diploid cells that are found in the testes and the ovaries that undergo meiosis. They produce gametes, and after meiosis we have sperm and egg cells that are haploid. In a human being, the diploid number is 46. That's 23 pairs of chromosomes. In the haploid gametes, the chromosomes aren't paired anymore, so there are just 23 chromosomes in the sperm, 23 in the egg. Now the sperm goes ahead and fertilizes the egg, and that egg or zygote will divide develop, the cells will differentiate, and what you'll wind up with are somatic cells. Those are the cells of the body, the diploid cells that make up the body tissues. Somatic cells are diploid, germ cells are diploid, gametes are haploid. What happens during meiosis? Meiosis is reduction division. Why reduction division? It's cell division that reduces chromosome number. Cells go from diploid with 2 sets of chromosomes to haploid with 1 set of chromosomes. The first step is DNA replication, which we see over here at number 1. It creates doubled chromosomes consisting of 2 sister chromatids. What's going on? Even though meiosis is reduction division, it starts in exactly the same way that mitosis starts, with a round of DNA or chromosome replication. So if you look at this cell over here, there are 4 chromosomes. If you look over here, there are still 4 chromosomes, but each one is doubled, consisting of 2 sister chromatids. In step 2, we have meiosis 1. And what meiosis 1 does is it separates the homologous pairs. So this chromosome is homologous to this one, this one, this one, and they're going to be separated. So now each of the resulting gametes only has 1 member of each homologous pair. That means it's now a haploid cell. But each chromosome is still doubled, and that's why we have meiosis 2. What meiosis 2 does is it separates the sister chromatids. The result is 4 unique haploid gametes. Compare mitosis and meiosis. Mitosis consists of 1 round of cell division that separates the sister chromatids. The cells begin as diploid and they end as diploid. The daughter cells over here are clones of the parent cell. It's used for growth and repair. Meiosis consists of 2 cell divisions. Meiosis 1 over here separates homologous pairs. So here we have the doubled chromosomes, homologous pairs. They get separated in meiosis 1. Meiosis 2 separates sister chromatids. We go from diploid to haploid. It's used to create gametes for reproduction. It introduces variation. The daughter cells are unique. How meiosis creates variation. I'm Mr. W from learn-biology.com, where we believe that interaction and feedback is what leads to deep, substantial learning. We're so sure of that we provide a money-back guarantee that comes with your subscription. What are the 2 ways that meiosis generates diversity? They're shown here. The first is independent assortment. The second is crossing over and genetic recombination. We'll explain both of those now. Explain what independent assortment is and how it generates genetic diversity. Note that the phases of mitosis and meiosis have the same names, the same designations. But because there are 2 cell divisions in meiosis, we have to give them a kind of suffix. So in meiosis, there's interphase 1 followed by prophase 1, metaphase 1, etc. Then there's a cytokinesis interphase 2, followed by a prophase 2, metaphase 2, anaphase 2, etc. The reason why that's important is because the events that we're going to talk about happen in between prophase 1 and metaphase 1. That's where independent assortment really takes place. What happens is that during prophase 1, homologous pairs pair up. And if you think about that, it's quite poignant and extraordinary. In the adult organism who's undergoing meiosis, the chromosomal parents wind up finding each other. And what I mean by that is that in the germ cells of an organism doing meiosis, the mother and the father's chromosome number 1 will find one another, and I'm not kidding, they actually embrace. And chromosome 2 does that, chromosome 3 does that. All those chromosomes find one another and embrace one another. During metaphase 1, they're pulled by spindle fibers, just as happens in mitosis, to the cell equator, right? So here we have that. But the way that each pair gets dragged to the middle is independent of every other pair. So in this very simplified system, it's possible that the paternal chromosomes might be on the left side and the maternal ones might be on the right side. It's equally possible that you could have this arrangement versus this arrangement, whereas you have paternal chromosome 1 on the left, maternal chromosome 1 on the right, whereas maternal chromosome 2 is on the left and paternal chromosome 2 is on the right. It's as random as flipping a coin, and that randomness is essential. What happens is that with two homologous pairs, 4 different chromosome arrangements are possible. That's 2 squared in the gametes. In other words, what we're going to do now during anaphase is we're going to pull these homologous pairs apart. So one possible arrangement is like this, and then in the gamete we have paternal chromosome 1, paternal chromosome 2, and in this gamete we have maternal chromosome 1, maternal chromosome 2. And if the chromosomes are organized like this, then this gamete can have paternal chromosome 1, paternal chromosome 2, and this gamete can have maternal chromosome 1 and paternal chromosome 2. Now, you can play around with this. You can cut out little pieces, or you can label coins M1, P1, M2, P2, and you can try different combinations, but you won't get more than 4 in a system that has 4 chromosomes as its diploid number. With 3 homologous pairs, then the math takes you to 2 cubed. That's 8 possible arrangements, and with 23 pairs, like we have in Homo sapiens, you have 2 to the 23rd possible arrangements. That's 8,388,608 possible combinations. That's the chance that any 2 sperm cells or any 2 egg cells would have exactly the same array of maternal and paternal chromosomes. So what's the chance that you and a sibling would have the same chromosomal inheritance? In other words, you'd inherit the same array of chromosomes in your dad's sperm and the same array in your mom's egg. Well, those are independent events. So the same egg, it's 1 over 2 to the 23rd. The same sperm, it's 1 over 2 to the 23rd. You multiply those two together, and that's 1 in 70 trillion. You ever wonder why you're different from your siblings? This is only one of the reasons why. That's why meiosis is so phenomenal. So this is independent assortment. What every chromosome does is independent of every other chromosome pair, and that creates tremendous diversity in the offspring. Independent assortment is a phenomenal engine for creating diversity, but there's yet another one in meiosis. That's crossing over. What is crossing over, and how does it create variation? When those homologous pairs pair up during prophase 1, they don't only embrace, they embrace in such an intense way that they actually exchange parts. So this embrace is called synapsis, and at a point called a chiasma, segments of DNA will move from one homolog to the other. The result is that you start like this. This array of 4 sister chromatids is called a tetrad, 4 tetrad. And so this is what it's like before crossing over, and then after crossing over, it's like this. Well, you'll notice this isn't really a maternal chromosome anymore. It's a maternal chromosome with a paternal piece, and the same thing for this one. So crossing over creates what's called recombinant chromosomes, and these have unique and novel sequences of DNA. How does sexual reproduction create diversity? One engine for diversity is independent assortment, and how it randomly arrays different combinations of chromosomes in the gametes. The other engine is crossing over and genetic recombination, which creates uniquely recombinant chromosomes. And then finally, there's fertilization, where the sperm and egg from different individuals combine in a fertilized egg. And that is the third generator. And that's why sexual reproduction creates diversity. It explains you, it explains me. Biology, is it amazing or what? Meiosis, the whole shebang. Let's walk through the entire process. We begin with interphase, and the thing to remember about interphase of meiosis is that it does exactly what interphase of mitosis does. It replicates the chromosomes, duplicates the DNA. That's why when we get to prophase 1, each chromosome consists of 2 sister chromatids. What else happens during prophase 1? The homologous pairs pair up. The maternal and paternal chromosomes find one another and they embrace. They do this thing called synapsis, and crossing over where they actually exchange pieces of DNA. So that's happening during prophase. During metaphase, the spindle fibers pull these homologous pairs to the center of the cell. And remember that each pair is pulled independently from every other. That's the source of one of the main sources of variation in meiosis, independent assortment. The way that these maternal and paternal chromosomes get arrayed is completely independent. It's 2 to the number of pairs if you want to mathematically calculate the number of chromosomal arrangements in the gametes. During anaphase 1, the homologous pairs are pulled apart. During telophase 1, a new nucleus forms. There's a cytokinesis 1 and an interphase 2 that's not shown in this diagram. That moves us to prophase 2, where the chromosomes once again condense. Note that whereas there were 4 chromosomes in prophase 1, there's only 2 in this cell in prophase 2. We've gone from diploid over here to haploid over here. That's the main transition, or one of the main transitions that occurs in meiosis 1. During meiosis 2, during metaphase, the doubled chromosomes get pulled to the cell equator and then they get pulled apart during anaphase 2. Then there's a telophase where a new nuclear membrane forms, and then there's another cytokinesis. The result, 4 haploid gametes, each consisting of single chromosomes. We've gone from diploid to haploid, from doubled chromosomes to single chromosomes, and each of these haploid gametes is unique. It's genetically unique, and that's what happens during meiosis. At learn-biology.com, we understand why students struggle with AP Bio. It's a hard course. The material is complex, the vocabulary is ridiculous, and the pace is withering. It's natural to feel overwhelmed and inadequate. To get an A or a 4 or a 5, you need an easier way to study, and that's why we created learn-biology.com. It has quizzes, it has flashcards, it has interactive tutorials about every topic in the AP Bio curriculum. It has a comprehensive AP Bio exam review system. Use learn-biology.com and you'll gain the skills and confidence that you'll need to ace your biology course and to crush it on the AP Bio exam. So here's your plan. Go to learn-biology.com. We've got free trials from June through March for both teachers and students. You won't believe how much you'll learn. Topic 5.6, Part 1, Chromosomal Inheritance, Sex Determination. How is sex determined in mammals? We've seen this image before. It's a karyotype. It shows all of the homologous pairs paired up. Chromosomes 1 through 22 are called autosomes. They're paired homologous pairs. They're the same in chromosomal males and chromosomal females. But the last pair are called the sex chromosomes. Females have 2 X chromosomes. Males have an X chromosome and a Y chromosome. Unlike the autosomes, chromosome pairs 1 through 22 in humans, the X and the Y are quite different from one another. For one thing, unlike those homologous pairs, they don't cross over and swap pieces of DNA. The X is a normal chromosome. It has a variety of alleles related to various non-sex relating functions. That includes immune function, vision, blood clotting, and so on. The Y has a region that's called SRY. It's indicated by this yellow bar over here. And that initiates the development of the testes during early embryonic development, and later on, production of testosterone, which winds up differentiating the body into its male form. During fertilization, it's the sperm that determines the chromosomal sex of the zygote, which becomes the embryo, which becomes the baby, which becomes the person. The males, which have their 22 autosomes and then an X and a Y chromosome, during meiosis will pass on either an X chromosome or a Y chromosome, because even though they're not truly homologous, they will get separated, just like how the other homologous pairs get separated. The egg has two X chromosomes, so every single egg cell is going to have the X chromosome. If the egg is fertilized by a X-carrying sperm, then the zygote will have two X chromosomes and it'll develop into a female. If the sperm that's carrying the Y chromosome does the fertilization, then the resulting zygote will be XY and it'll develop into a male. That's chromosomal sex determination in all of the mammals. The fact that half of the eggs will be fertilized by an X-carrying sperm and half will be fertilized by a Y-carrying sperm explains the fact that among the births in any mammal population, the initial ratio of males to females will be 50-50. Birds also have a chromosomal system of sex determination, but it's different. It's kind of a flipped version of the one in mammals. In birds, it's the egg that determines the chromosomal sex of the offspring. That's because the females have what we call a Z chromosome and a W chromosome, and during meiosis, half of the eggs will wind up being Z and half will be W. The females will pass on either that Z or W chromosome. The males have two Z chromosomes, so when they form their sperm, all of the sperm carry Z. Fertilization of a Z-carrying egg will lead to a male who's ZZ. Fertilization of a W-carrying egg will lead to a female who is ZW. That's chromosomal sex determination in birds. As in mammals, because half of the eggs will be Z eggs and half the eggs will be W eggs, the result is that among the initial births that happen in any bird population, half of those birds will be male and half of those birds will be female. Among certain reptiles, sex is determined by the temperature at which the embryos develop. Some reptiles lay their eggs in a nest that's dug in the sand. And as you might imagine, it's warmer on the top, closer to the sun, and cooler down below. These animals develop based on a pivot point that's represented by TPIV over here. So in sea turtles, the eggs that develop above the pivot point become female. Here's the proportion of males over here. So if you're above the pivot point, if you're in the warmer area, you'll develop into a female. If you're in the cooler area over here, below the pivot point, you'll develop into a male. And then at the pivot point, it's pretty much random, 50-50. In another kind of reptile called a tuatara, it's the opposite. So basically, if you develop above the pivot point, then you have a higher chance of being a male. If you're below the pivot point, then the proportion of males goes down. You have a higher chance of being a female. And in crocodiles, there are two pivot points, a low temperature one and a high temperature one. In the coolest and the warmest temperatures, the eggs develop into females. In other words, the proportion male, way down. But at intermediate temperatures over here, the eggs develop into males. How is sex determined in ants, bees, and wasps? This is completely mind-blowing. It's a system that's called haplodiploid sex determination, or haplodiploidy. The males are haploid. They develop from unfertilized eggs. So here's a male here, haploid. The females, which include the queen and all of the workers, they're all diploid and they develop from fertilized eggs. So the mother in a bee colony, just to use that example, is the queen. She undergoes normal meiosis when she creates her eggs. But the father is a haploid male, also called a drone, and he can't really do meiosis because he's haploid, he's not diploid. So essentially he passes on 100% of his chromosomes in the sperm that he creates. The consequence is that all the bees in a hive, with the exception of the drones, all of the worker bees are sisters, but they're more closely related to one another than any two mammal sisters are. Just think about it. Mammal sisters inherit half their mom's genes, half of their dad's genes, so they're 50% related to one another, whereas these sisters inherit half of their mom's genes and 100% of their dad's genes, so they're 75% related to one another. They're more closely related to one another than they would be to their own offspring. So that is thought to be an explanation of why the workers cooperate with one another to help the queen create more offspring, to keep the hive going, to find food, all of these incredible foraging behaviors that you find in ants, wasps, and other social insects. But it's not a complete explanation because, like, for example, the termites are also social, but they don't have this haplodiploid system. So biology, amazing. This is as much as you need to know about haplodiploidy for the AP Bio exam. Topic 5.6, Part 2, Chromosomal Inheritance, Non-Disjunction and Chromosomal Variation. What is non-disjunction? What are its consequences? Non-disjunction is kind of a cool word. So a junction is where things come together. A disjunction would be things coming apart, and non-disjunction means things failing to separate. It's when the homologous pairs or the sister chromatids don't separate during meiosis. There's a couple of variations. In meiosis 1, the homologs don't separate, and as a result, so you see that over here, this blue chromosome over here, these homologs didn't separate, they stayed together. So the result is that in meiosis 2, we're going to have 3 chromatids on this side, 3 over here, 1 over here, 1 over here. So 50% of the gametes are N haploid plus 1 extra, and 50% are N minus 1. So N, the haploid number, missing a chromosome. If non-disjunction occurs during meiosis 2, it involves the sister chromatids not separating. In meiosis 2, the sister chromatids don't pull apart. So the result is that 25% of the gametes are N plus 1, 25% are N minus 1, in other words, the haploid number but missing 1, and then 50% of the gametes will be normal. So that's what non-disjunction is and how it can happen in meiosis 1 or meiosis 2. We saw in the last slide how non-disjunction during meiosis 1 or meiosis 2 can result in gametes that have an abnormal number of chromosomes. If the eggs are N plus 1, they have the haploid number plus 1 more, then the zygote will have an extra chromosome. Then what we have is a trisomy. And what that means, tri is 3. Instead of a homologous pair with 2, we have 3. The most famous example of that is Down syndrome, which is a trisomy of the 21st chromosome, which has various developmental consequences and developmental delays. If the eggs are N minus 1, like over here or over here, and the sperm again are carrying a normal number of chromosomes, so that's not necessarily the case. You can have non-disjunction that occurs during the formation of sperm as well, but then the zygotes will have a missing chromosome. So they'll have all of the homologous pairs, but one will be short. And the result of that is a monosomy, and most of those aren't really survivable except in the case of the sex chromosomes. So one to know is called Turner syndrome, where instead of females having 2 X chromosomes, they have 1. And there's a significant amount of variation that could happen in the sex chromosomes. There can be men who are born with an extra X chromosome. There can be men who are born with an X and 2 Y chromosomes. So those are all chromosomal variations that come about through non-disjunction followed by fertilization. I want to acknowledge how difficult and complex some of these concepts can be, and I want to encourage you to go to learn-biology.com, and with a free trial you can do the tutorials and you can use our unit reviews, and it's going to really help you to get on top of this material, setting you up for success on your unit test or the AP Bio exam. Topic 5.3, Mendelian Genetics. Genes are the basic unit of heredity. They are what gets passed from parents to offspring. They determine traits or characteristics. You can also think of genes from a molecular genetics perspective, and you can think of them as a sequence of DNA nucleotides that code for RNA and ultimately code for protein, and those proteins ultimately determine the characteristics of the organism. Describe Mendel's principle of segregation. In your answer, cover the difference between the terms homozygous and heterozygous. Just a historical note, Gregor Mendel is considered the father or grandfather of genetics. In the 1800s, he figured out many of these basic principles that we're talking about right now. Every individual has 2 copies of each gene. Those copies are located on the chromosomes that are organized into homologous pairs. The alleles are alternative versions of those genes that might have different DNA sequences that will produce proteins that have different amino acid sequences. Homozygous means that the 2 alleles are identical. For example, in this parent over here, both of the alleles are designated with a capital letter A. They're the same. This person is a homozygote for this particular gene. In a heterozygote, the alleles can be different. In this individual over here, one of the alleles is capital A, and one of the alleles is small a. The principle of segregation shows what's happening here. When individuals create gametes, their sex cells, they pass on only one of their 2 alleles. So the alleles are together in the parent, but they become segregated or separated during gamete formation. Here that's happening in the formation of sperm. Here that's happening in the formation of eggs. If you feel that that corresponds to the events of meiosis, you're onto something, and we'll talk about that later. Define the terms dominant and recessive. Dominant alleles are always observed in the phenotype of the offspring. They're represented by a capital letter. So, for example, the capital letter A. So here's an individual who's homozygous dominant, and they express the dominant characteristic, dark black fur. In these heterozygotes, there's also a recessive allele, but because the dominant allele is present, then the characteristics of the organism is determined by the dominant allele. A recessive allele only shows up in a homozygote. So this mouse over here is homozygous recessive, and therefore it has the recessive appearance. That homozygous allele recedes into the background when it's paired with a dominant allele, and these are represented in the Mendelian system by a lowercase letter, in this case, lowercase a. What's the difference between genotype and phenotype? Phenotype is your appearance. It's the observable characteristics in an organism. An example is brown eyes. Genotype is the genes that you've inherited, the type of genes that you have. It's your underlying DNA. In my case, my eye color is brown. That's a dominant phenotype, though eye color is actually complex. There are many alleles involved in determining eye color. Actually, I think it's about 3, but I'm a heterozygote. So my genotype would be like capital B, little b. How do I know that? Because my wife has blue eyes and both of my kids have blue eyes, and that would only be possible if I were a heterozygote, because if I was a homozygote, then I could only pass on the big B allele, and then both of my children would have been heterozygotes. Explain what a monohybrid cross is and what the results of such a cross will be. In your response, create a Punnett square to demonstrate your understanding. This, which we've seen before, is a Punnett square. It uses the rules of probability to quickly predict the offspring of genetic crosses. A monohybrid cross is a cross between 2 heterozygotes. So these are peas, which was one of Mendel's study organisms, and this pea has purple flowers. The genotype is capital P, little p, capital P, little p. Why? Because it's a heterozygote, and this is a monohybrid cross. So they're hybrid for one characteristic. Now, the result will be offspring in a 3-to-1 ratio. So how do you do these Punnett squares? Well, you start by identifying the genotype of the parents. Well, in the problem, it's given to you. It's a monohybrid cross, big P, little p, crossed with big P, little p. So you can think of this as a germ cell. It's going to go through meiosis. The homologous pairs are going to separate, principle of segregation. So some of the gametes will have big P, some will have little p. And that's true for each parent here, big P, little p. Flowers are hermaphroditic, so it doesn't really make sense to talk about father and mother in this case. What you do is then the gametes will fertilize one another. You know, if this were a pollen, the equivalent of sperm, it would fertilize the egg, assuming this is the female and this is the male over here. But in other words, this combines with this, and we have this organism is capital P, capital P. It's got the dominant phenotype. It's homozygous dominant. This organism gets a big P and a little p. It's a heterozygote, and it still has the dominant phenotype. Same with this one over here. This one gets two little p's, one from each parent, and as a result, it shows the recessive phenotype, and that recessive phenotype is white flowers. We have three individuals with the dominant phenotype, one with the recessive phenotype. A quarter of the offspring are homozygous dominant. Half of the offspring, 2/4, are heterozygous. They still have the dominant phenotype. And 1/4 will be homozygous recessive, and they'll show the recessive phenotype. That's how you do a Punnett square for a monohybrid cross. In genetics, what are the P, F1, and F2 generations? This is a common notation that's used in genetics, and you, as an AP biology student, need to understand it. The P generation is the parental generation, and in general, they're true breeding homozygotes. Now, notice that we've jumped in complexity. In this example, we're not dealing with just one gene, we're dealing with two. So there's a gene for tallness, and there's the gene for flower color. And the alternatives are tall versus short, and purple flowers versus white flowers. When you breed the P generation together, those offspring are called F1s. It's the first filial generation. And in this case, they're heterozygotes. They're double heterozygotes because they're big T, little t, that's a heterozygote, and big P, little p, so double heterozygotes. Now, let's go from the F1s to the F2s. You breed together the F1 generation, these double heterozygotes, and you get the F2s. So the F1, that's the first filial generation. And the F2, it's the second generation. It's the grandchildren of the P generation. And crossing the dihybrid F1s. Why are they dihybrid? Because they're double heterozygotes. They're hybrid. They're mixed for two characteristics. That's called a dihybrid cross. We're going to take that apart in just a moment. Define Mendel's principle of independent assortment. Independent assortment, it's what we talked about in the context of meiosis. This is essentially the same process. Mendel was able to intuit this just looking at the transmission of traits from one generation to the next. Genes carrying different traits are segregated and passed on independently from one another. So in an organism that's dihybrid, big T, big P, little p, the way that the T gene pair gets passed on is independent from the way that the P gene pair gets passed on. And therefore, in a double heterozygote, a dihybrid, big T, big P, little p, 4 unique gametes can be created. Here they are. Big T, big P, big T, little p, big P, little t, little p. How can you figure this out for yourself? It's very, very simple if you use the FOIL algorithm. That's for factoring binomials. You should know it from algebra. If you don't, just remember it. First, outside, inside, last. So you think about this and you think about the gene pair. So you take the first one, big T, big P. You take the outside one, big T, little p, that's over here. Then you take the inside one, little t, big P, that's over here. And then the last one, little t, little p. So 4 gametes can come from a dihybrid organism. This is limited to genes that are found on different chromosomes. The game completely changes when those genes are on the same chromosome, and we'll deal with that later. Explain what a dihybrid cross is and what the results of such a cross will be. It's a cross between two double heterozygotes. A couple of slides ago, we were looking at the production of the F2s when there was a dihybrid cross. We're crossing big T, big P, little p with the same. You're going to use the FOIL algorithm to figure out the gametes, and we just described that in the last slide. You just bring these alleles down and over and combine them. You do that for all 16 squares of this dihybrid cross, and what do you wind up with? You wind up with a 9:3 to 3:1 ratio in the offspring, and that is actually something that's worth memorizing. What's the connection between Mendel's laws of inheritance and what happens during meiosis? We've just articulated Mendel's two laws, the principle of segregation and the principle of independent assortment. The principle of segregation, according to Mendel, says that parents have two alleles for each trait, but pass on only one to their offspring, which inherit their two alleles from two separate parents. In meiosis, there's a diploid parent that produces haploid gametes. The diploid phase corresponds to the two alleles in each parent. The haploid phase corresponds to the one allele that the parent passes on to their offspring. In zygotes, that diploid condition is restored, and that's not shown in this diagram over here. Independent assortment, according to Mendel, is what happens to one gene pair is independent of every other gene pair, at least in the ones that Mendel studied. In meiosis, chromosomes assort independently during metaphase 1 of meiosis. What is the rule of multiplication? Demonstrate your understanding by explaining how to predict the probability of genotype little a, little b, little c, resulting from a trihybrid cross between big A, big B, big C, little c times the same. Don't make a huge Punnett square to try and solve problems like this. What you want to do instead is use one of the laws of probability, the rule of multiplication. Here's what it is. The probability of independent events occurring together is the product of their individual probabilities. Here's how to think about this. It's like three independent Punnett squares, because what each gene does is independent of every other gene. What A is going to do is independent of B, is independent of C. So three independent Punnett squares, one for A, one for B, and one for C. But you don't have to create three Punnett squares. You just have to use the rule of multiplication. In a cross between big A, and big A, little a, the probability of little a is 1 out of 4. It's the same for B, 1 out of 4, and it's the same for C, 1 out of 4. These are independent events, so you use the rule of multiplication. It's 1/4 times 1/4 times 1/4, or 1 out of 64. Topics 5.4 and 5.5, Non-Mendelian Genetics and environment-phenotype interaction. Non-Mendelian genetics is about genetic principles that were discovered after Mendel's original contributions. One of the most important of these involves linked genes. What are linked genes? Describe their inheritance pattern and explain which Mendelian rule linked genes don't follow. Linked genes are genes that are on the same chromosome. So fruit flies, as opposed to peas, were the widely used experimental organism to discover the principles of non-Mendelian genetics. And here you can see one chromosome from a fruit fly, and there's a whole variety of genes, sequences of DNA that code for specific traits that are located on the same chromosome. One has to do with this kind of bristled appendages on the head. One controls body color, one controls one type of eye color, one controls wing length. These genes are mostly inherited together, which is different from the independent assortment that we saw with Mendelian genetics. Because they're on the same chromosome, these genes don't independently assort. So like, for example, above, genes T and A in this cell over here, they're linked. In this cell over here, they're not linked. These would independently assort, and these wouldn't. So what happens in crosses involving linked genes? Note, first of all, that we have a different symbol system over here. You can see that right over here. We have B plus, VG plus, VG plus. In this system, in non-Mendelian genetics, a plus sign indicates the wild type or the dominant allele. If you have a symbol that can be more than one letter without a plus sign, that indicates the recessive. In this P cross, what we're doing is we're crossing normal body, normal winged fly, B plus, VG plus, VG plus, with a black-bodied vestigial winged fly. Those are both recessive traits. And note that all the F1 offspring are dihybrid. They're B plus B, VG plus VG, and they have both dominant phenotypes. This is what you'd expect in a Mendelian trait for the F1s. We have a gray body and normal winged fly, which has all of the dominant characteristics. Now over here, what we're doing is we're representing this chromosomally. B plus, VG plus, and BVG. Notice that B plus and VG plus, they're on the same chromosome, and BVG are on the same chromosome too. These genes are linked. What if they were perfectly linked and they never separated? What would you expect to happen? The method here is a little bit different than the method in a Mendelian cross. We're not doing a dihybrid cross, we're doing what's called a test cross. And a test cross, a dihybrid, so B plus B, VG plus VG, is being crossed with a double recessive. And the way that it's done is the double recessive is the male, the female is the double hybrid, the double recessive. I've represented this over here. Here's a representation of the female. On one of her chromosomes, she'll have B plus and VG plus. On the other chromosome, she'll have B and VG. The male is a double mutant, and he has B and VG on both of his chromosomes. When the female produces gametes, then half of her gametes will have a B plus and VG plus, and half of her gametes will have B and VG. Now that's assuming perfect linkage. In the male, all of the sperm have to have B and VG. Here's a Punnett square. These are the eggs, that's going here. The other eggs are going here, the other half of the eggs, and all of the sperm are going over here. You put them together, and what you'd expect is that half of the offspring would be B plus B, VG plus VG. The other half of the offspring would be B, B, VG, VG. This organism, these offspring would have normal body and normal wings. That's 50% of the offspring. And the other 50% of the offspring would have black body and vestigial wings. But that is not what happens. And what I'm doing here is I'm letting you know what you would expect if there were perfect linkage. What actually happens in a cross involving linkage? Note that the numbers won't always be the same, but the general concepts apply. What we see is that the majority of the offspring have parental phenotypes. What does that mean? Well, the mom's phenotype was gray body with normal wings, and many of these flies have that phenotype, gray body, normal wings. The father's phenotype is black body with vestigial wings. Many of the offspring have that phenotype, but a significant number of offspring have recombinant phenotypes. What are recombinant phenotypes? They take one of the phenotypes of the mom and they combine it with one of the phenotypes of the dad. That's what's happening over here in these flies that have a gray body like the mother and vestigial wings like the father. Or you can have a black body like the father with normal wings like the mother. Those are recombinant phenotypes. Why do we have most of the offspring having parental phenotypes, but a significant minority having recombinant phenotypes? It's because linkage is not perfect. Genes that are linked don't always stay together. Why not? Because during meiosis, there's recombination and crossing over. So linked genes, because of that process, can separate. We'll see the details of this in the next slide. What happens in crosses involving linked genes? Why are there recombinants? In diagrams A and B, we have a dihybrid female and her germ cells. In C, we have meiosis 1 with crossing over. We have homologous pairs. They're going to get together. They're going to swap genes and the production of recombinant chromosomes. But notice that some of the sister chromatids are recombinant and some aren't. So now what we're going to do is we're going to complete meiosis. We're going to go through gamete formation. And when we do that, note that of the eggs that the female produces, 2 of them at letter H, they're recombinant. What do I mean by that? Well, they have B plus and they have VG. In other words, this chromosome is B plus VG plus, this chromosome is B VG. Well, the recombinant ones are the ones where those alleles swapped places. So this is a recombinant B plus VG, this is a recombinant B VG plus, but these 2 at letter I, those are parental, right? They're just like the parents, B plus VG plus B VG. Now over here, we have the male parent who's a homozygote, and his gamete, he can only produce one kind of gamete. He's homozygous. He can only produce a gamete that has B and VG. Here's what happens with the offspring. Offspring N and P have recombinant phenotypes. In other words, they have, in this case, gray body vestigial wings. That's recombinant. Neither of the parent look like that. And this is black body normal wings. Again, recombinant. Neither of the parent look like that, but O and Q are parental phenotypes. O is gray body normal wings and Q is black body vestigial wings. Why are there so many more parentals than there are recombinant phenotypes? It's because crossing over happens, but it happens at a rate that's dependent on the distance of these alleles on the chromosome. And so the closer 2 alleles are, the less they'll tend to cross over. And this frequency, which is, I believe, 17%, that represents the distance between the B allele and the VG allele on the fruit fly's chromosomes. What's the relationship between the percentage of recombination and the distance between any 2 linked genes? The further apart any genes are on the chromosome, the higher the percentage of recombinant gametes. In the chromosome map above, genes A and E will recombine the most because they're the furthest apart. In other words, there can be crossing over here, over here, over here, and all of those will get E to jump over here and big E to jump over here. That's going to look like recombination. That's going to actually almost look like independent assortment because they're so far away. But genes B and C, they'll recombine the least because the only way that they can cross over is if there's a chiasma right over here at this specific spot that would enable little c to jump over here, big C to jump over here, or B could do the same. The percentage of recombination can be used to calculate the map distance between 2 alleles. So over here, this is a chromosome map, and it's saying here that the distance between this long bristled appendages called long aristea and gray body, that's 48.5. Well, those are units that reflect the frequency of recombination. Over here between gray body and red eyes, that's a little bit under, that's 9 recombination units. And really what this amounts to is the frequency of recombinants. So doing all of these mapping experiments or doing all of these breeding experiments with fruit flies, the researchers at Columbia University in the 1900s, Thomas Hunt Morgan and his crew, were able to create maps of chromosomes, and this was establishing that genes are on chromosomes and was part of the pathway that ultimately led to the double helix and all that other great stuff. Note that these are not physical maps so that the numbers don't add up, but this should get you far enough to have a basic understanding. Again, on learn-biology.com, I have a whole tutorial about linkage and recombination with practice problems that'll get you all the way there. Your success in AP Biology starts here. Are you struggling with AP Bio? With learn-biology.com, students get the skills and confidence to be a top student and earn 4s and 5s on the AP Bio exam, guaranteed. Go to learn-biology.com to find out how you can master your biology course and crush the AP Bio exam. What are sex-linked genes? Sex-linked genes are located on the X chromosome. Males, because the genes are located on the X chromosome, can't be heterozygous. They either have the allele or they don't. So the whole heterozygous, homozygous thing doesn't apply to males. But females can be heterozygous, as shown. In this case, it's X big H, X little h, a heterozygote. or they could be homozygous dominant. That would be X big H, or they could be homozygous recessive, X little h, X little h. Using hemophilia as an example, how can males inherit a recessive sex-linked trait? Hemophilia is a blood clotting disorder, and essentially hemophiliacs can't clot their blood. It's much more common in males than it is in females because the allele for hemophilia, the mutated allele that leads to ineffective blood clotting, is on the X chromosome. Sons inherit their X-linked alleles from their mothers. So we'll look at the Punnett square first. This Punnett square is from learn-biology.com. These boxes are things that you would drag the alleles into. Here we have a hemophiliac, X little hy. It's a recessive X-linked condition. How did this young man inherit that X-linked allele? From his mom. Mom is a heterozygote, X big H, X little h. She's not a hemophiliac, but she carries the allele. It's commonly known as a carrier. The dad, in this case, is unaffected, X big HY, but it doesn't matter because dads don't pass on their X chromosomes to their sons. They pass on their Y chromosomes. That's why they're sons to begin with. So the mom has to be a heterozygote, as shown here, or she could be homozygous recessive, though that's quite rare. We'll look at that in the next slide. Here's a pedigree that shows the same thing, basically a cross between a heterozygous female and a normal male. And note that in this case, the mom passed on her normal chromosome, and the dad passed on his normal chromosome. It's the only X chromosome that he has to pass on. In the case of these two sons, the mom passed on her defective X chromosome with the hemophilia allele. So we have two sons who are X little hy. They're both hemophiliacs. This daughter over here is a carrier. She received the normal X chromosome from her dad and an X chromosome with the mutant hemophilia allele from her mom. And here we have a son who inherited the normal X chromosome from the mom. She had one of two to give. He got lucky, and the Y chromosome from the dad. So he's a normal young male. Examples of X-linked recessive conditions in humans, hemophilia, which we just talked about. Also, a very common one is red-green color blindness in fruit flies, which have a similar sex determination system to humans. It's just the X chromosome and the Y chromosome, just like mammals. The allele for white eyes is a mutation that's on the X chromosome in fruit flies. And in fact, this was the first allele that was ever located on a specific chromosome. Can a female inherit a recessive sex-linked trait? Absolutely, but it's uncommon. Here's what has to happen. The male parent must have the sex-linked recessive trait. So here we have a wide-eyed fruit fly, X little WY. The female must be a heterozygote in this case, or have the trait. So it could be a wide-eyed female. Here the female is X big W, X little w. And when we create the Punnett square, we can see that among the female offspring, 50% of the offspring are carriers. They have to be carriers. They get the normal allele, for red eyes in this case, from their mom. They get the recessive allele, for white eyes, from their dad. And in this case, they get the recessive allele from both parents. So 50% are carriers, and 50% of the females have the trait. Among the male offspring, 50% of the males are normal. Why? Because the mom's a heterozygote. She passes on her normal X chromosome with the red-eyed allele to half of her sons, but there's also the 50% chance that she'll pass on the recessive mutated allele, and that's why this offspring over here will be white-eyed. But the key thing that we were looking for was, how can a female wind up inheriting a recessive sex-linked condition? She can if the dad has the condition and the mom is a heterozygote. What is non-nuclear inheritance? I'm Mr. W from learn-biology.com, where we believe that interaction and feedback is what leads to deep, substantial learning. We're so sure of that we provide a money-back guarantee that comes with your subscription. It's inheritance of genes that are not on a nuclear chromosome, but on the chromosome of a mitochondrion or a chloroplast. A nuclear chromosome is one of the autosomes or a sex chromosome that are found inside the nucleus of a eukaryotic cell. These genes that are on mitochondria or chloroplasts are only passed on to the offspring through the female gamete. Why? Let's look at the example of sperm. Sperm have mitochondria. The mitochondria power the flagellum that enables the sperm to swim towards the ovum. But when there's fertilization, only the head of the sperm penetrates through the egg membrane or is allowed in through the egg membrane, and all of the mitochondria are left outside. So all of the mitochondria that get passed on to the offspring are mitochondria that were passed on through the female germ cell. The inheritance pattern of these mitochondrial or chloroplast genes doesn't follow Mendelian patterns. You can see that in this pedigree over here, where the mom passes on the mitochondrial gene to all of her offspring, male and female. But the male who inherits the mitochondrial gene doesn't pass it on to any of his offspring. The male is essentially a mitochondrial dead end, if we're talking about mitochondria, where the female passes on her mitochondria to all of her offspring. In the case of mitochondrial inheritance in eukaryotes, all of one's mitochondria are inherited from one's mother. And in plants, they also have mitochondria, so the same rule applies, but they also have chloroplasts, and only the ovule, not the pollen, passes on chloroplasts and mitochondria to the offspring. So it's the same thing. It's female line inheritance of alleles when it relates to genes that are on an organelle, such as the mitochondria or a chloroplast. What is incomplete dominance? This is when the phenotype of the heterozygote is different from and usually intermediate between either of the homozygous phenotypes. Neither allele is dominant. So in this case, we have to use a different notation system. This is carnations, and C big R with a superscript represents the red flower color allele. C big W with a superscript represents the white flower color allele. So here's our P generation, C big R, crossed with C big W. The F1 generation, they're all hybrids, C big R, C big W, but they're pink. It's because 2 doses of gene expression, in other words, this is DNA that's coding for protein, and you need that much protein in order to produce that vibrant red color. If you just have one, then it's not enough to push the phenotype all the way to red, and you wind up with this intermediate phenotype. One dose can't produce sufficient pigment to create the red color. Let's see what happens when you do your F1 cross to produce the F2 generation. So we have a pink carnation, C big R, C big W, crossed with another pink carnation, C big R, C big W. What do you get in the offspring? You get one offspring that's red because it's C big R, one offspring that is white because it's C big W, and 2 that have the intermediate phenotype pink because they're going to be C big R, C big W, C big R, C big W. Explain how the same genotype can result in different phenotypes under different environmental conditions. This is also known as an environment-genotype interaction. And what happens is that factors in the environment influence gene expression, and that leads to variation in the subsequent phenotype. And basically what it's saying is something that's fairly well known, that genes don't determine everything. Genes interact with the environment. And note that this is the norm, not the exception. Here are 2 examples to know about. If you have hydrangeas in your yard, you can determine the color of the hydrangeas because the flower color is determined not by genes but by the acidity of the soil. In a more acidic soil, shown over here, you'll get this beautiful purple color. In more alkaline soil, you'll get reddish color, and there's actually mixes that you can add to the water that you give to your hydrangeas that will adjust the alkalinity or acidity of the soil. Example number 2 is dark fur regions in mammals. and they develop in cooler body regions away from the core. This is a Himalayan rabbit, but if you can picture a Siamese cat, it's pretty much the same thing. So this is a cool region, this is a cool region, this is a cool region, and all the cells have the same genes, but the cells that are being exposed to cooler temperatures express different pigment alleles than those that are in the warmer regions of the body. There are many other examples. Height and weight in humans is caused by an interaction between the genes that someone inherits and the environment that they're in. The way that your skin color can change in relationship to the amount of sun that you're exposed to. That is genes being expressed in relationship to environmental cues. We talked earlier about how sex is determined by temperature in certain reptiles. Another example of environment genotype interaction. Here are your next moves for AP Bio success. Please subscribe to learn-biology.com and please watch this next video.